All right, everyone. Good morning. Welcome all to the 2024 Interagency Ecological Program Annual Workshop. Thank you all so much for being here. I'm so glad that we could all be here in person again in our beautiful Natural Resources Agency headquarters building, beautiful auditorium here. So good to see people in person and, have the option for people at home who can't be here to see what we've got going on today, because we've got an excellent program. And I'm very excited to showcase all the hard work that IEP has done over the past year. Especially, given the state's budget crunch, all sorts of things. There was a few minutes we weren't sure this would happen, but we did manage to pull it off. So we're very, very excited about it. A couple of logistical things before we get started. First of all, if you are a presenter today and you have not loaded your talk onto this computer, please stay for a minute after the break to get your talk loaded up. We want to make sure they're all here so that the day goes smoothly. If you haven't seen, there are QR codes posted around with links to the agenda, as well as the main workshop page, a booklet of all of the abstracts, and more information about lunch in the surrounding area and stuff like that. Bathrooms are out in the lobby around the corner. If you are working in our overflow room, we do have an overflow room up next to the poster session, which is upstairs and across the way. If you're working there or need to step out and check your email, the Wi-Fi code is CNRA guest, CNRA all capitals, and the G in guest is capital. In our auditorium, it's a new building. It's very nice. So please do not eat or drink anything besides water in the auditorium. We do have coffee that has been sponsored by ICF. Huge thank you to ICF for sponsoring the coffee. Science runs on caffeine, but please enjoy that coffee in the lobby. If you are here in the auditorium, you can have water in a water bottle or a travel mug. So if you're drinking anything out of a travel mug, I'm assuming it's water. If anyone needs any special accommodations, help getting upstairs to the poster hall, anything like that, or extra hearing devices, please see Christine outside in registration. So this year, we've had a lot of changes in IEP. We always seem to have a lot of changes, but IEP remains an excellent group of scientists who do amazing work. We've had some programs, long-running programs, close out this year, as well as starting new programs or expansion of programs. And this week's program reflects the diversity of studies, both new and old, that we've seen at IEP. This program also includes not only ecological sciences, but also some social sciences and policy topics. We even have art created by community members on display in the poster hall as well. This year, we've tried to really elevate early career scientists and students in our program. So there is as usual, an award at the end for the best early career presentation and early career poster. The workshop is hybrid, so while we'll take questions from the audience, both in person and online, please know that the workshop is going to be live streamed and most of the presentations will be recorded for later watching. So if you are saying anything, know that you are potentially on camera. The other highlight to this year's program that I want to point your attention to is back after a brief hiatus. We will have a poetry slam tomorrow before the second poster session. This is a chance for poster authors to give a brief poem, skit, song, whatever, to sort of plug their poster, tell people why they might want to visit it. Or if you just are inspired by the beauty of the ecology of the Delta and have a brief poem, song, or skit that you would like to present, it'll be an open mic. We'll just have people line up and present their their poems. So please, I encourage everyone to participate. It's usually a lot of fun. If you're not feeling inspired, you're not a poet, but would like to do it anyway, come see me and I'll write you a limerick. Finally, before we start the program off for real, I just want to give a huge thank you to the organizing committee who put together this program, all of the session chairs who have stepped up to help organize the sessions and introduce the presenters, all of the volunteers. We have many volunteers running mics, keeping time. In the lobby, we have external volunteers from the Natural Resources Volunteer Program to assist with registration, so all of you can watch this instead of being out there, and up in the overflow room. And especially a huge thanks to Christine, who we can't even applaud right now since she's outside still working hard, but huge round of applause to everyone who helped put this conference together. And with that, I am going to give it over to our first plenary speaker of the day, Steve Culberson, your IEP lead scientist, who is going to give us an overview of 10 things he learned in IEP.
I do that before I take off my glasses because now I can't see. Good morning and welcome to the IEP Annual Workshop, the 2024 edition. Thank you to Rosemary, Hartman, and Christine Job and the Workshop Planning Committee for putting together yet another outstanding program for our stimulation, communication, and education. It's been another reflective year for me, and I've been thinking about 10 things I've learned working in the IEP. These are my impressions, and hopefully they'll be useful for framing our dialogues as we travel over the next 3 days together. These will be necessarily short and sweet in nature, and perhaps only mildly fleshed out, but they do come as the result of long hours of trying to guide the IEP science in ways we might all eventually agree will be beneficial to the largest constituency possible. So in no particular order, constant dialogue is fundamental. Creating useful technical information from the estuary for decision makers must involve continuous dialogue between those who make decisions and those who make environmental observations. Of the impediments to implementing good policy informed by good science, over my career, the tendency to fall back on throwing a draft or final research project over the wall for decision makers to read has been one of the most common and enduring impediments in the policy science realm. Likewise, the one-off meetings we have wherein decision makers tell the technical folks what they need in order to make their decisions, expecting that meeting to result in research program marching orders, nearly always results in misguided research, as cutting edge, peer-reviewed, and peer-esteemed as it might be. Far more effective in my experience have been projects that included close communication and transmission of information via documents and in-person meetings and learning, co-produced workshops over the long term, and as a requirement of implementation. Unfortunately, this design and this extent of interaction remains the exception rather than the rule, since many decision makers have too much responsibility to pay the right amount of attention to any one thing in their portfolios. I think this is in itself another problem. We tend to hire people as bodies rather than as experts or as knowers, but that discussion will have to wait for another sermon. I do touch a little bit more on this in point number 5 below. But in any event, the science policy conundrum we face should be seen as a human conversation, not as a commodity-based transactional interaction. The more we invest in our conversations, the better our science policy interface will be. Near-term knowledge requires long-term context. Credible understanding of near-term change requires long-term monitoring, trend detection, and analysis. And I call your attention to one paper by Enright and Culberson, 2009, for one example of this perspective. There are, of course, many others. Using harmonic deconstruction analysis techniques, we understand now how a 70-year data record of salinity field measurements within the San Francisco estuary have distinguishable temporal features spanning from minutes to days to decades in length. Only once we knew what the longer term, in this case 70-year record was telling us, could we discern properly the shorter term characteristics and understand their full meaning given tidal harmonics and seasonal, annual, and decadal weather pattern forcing and processes. Without the proper long-term context, we wouldn't know what we were looking at. For example, a local maximum or a global minimum. Nor would we know what direction any trend was heading. Was it up, down, or was it just noise? In the words of the authors, quote, "We underscore the value of long data records for discerning trends and variability drivers. On the one hand, ocean atmosphere climate teleconnections explain significant decadal scale, 20 to 25 years, variability. On the other hand, slowly changing watershed runoff characteristics, northern reach bathymetry deepening, and expanding water project operations together explain long-term, greater than decadal trends. Therefore, identifying trends and mechanisms requires data sets that are longer than the scale of the lowest frequency forcing mechanism." In other words, for the IEP, keep collecting environmental data. You don't know when you'll be in the dark without it. Number 3. Measures of central tendency are inappropriate for assessing ecology. Measures of and analysis using techniques based upon measures of central tendency are to be viewed with caution and skepticism when understanding and managing ecosystems in the long term. Manufacturing processes and prescribed treatment and effect experimentation are appropriately studied using traditional frequentist statistical assumptions. Principal among those assumptions is the idea that a system under scrutiny is the notion that the system under scrutiny tends towards some average state, identifiable as and characterized by a system demonstrating a measure of central tendency. In contrast, in my view, biology and ecology do not do this. They do not trend toward the middle. Organisms and ecosystems, as DNA enablers, if you will, reward bet hedging, risk-taking, extreme behavior, or genetic-based variations in buffering against those conditions. Ecology and organisms require perturbation and disruption to evolve and persist, not stasis, not the mean, or say the average streamflow condition. Biology finds opportunity in change, in metamorphosis, in maturation, in learning. I'm increasingly disappointed in our lingering physics envy and its legacy versus understanding ecosystem and organisms on their own terms. Some legacy of our biological presence, will persist on this planet long after our fancy mathematical models have perished. Our inherent understanding of our place in this world and our ability to exist within it is in our recovered ability to see biology as biology and not as mathematics, as statistics, or engineering. We are biological beings, messy, unpredictable, chancy, emotional. Get over it. Have a feel for what you are attempting to manage. If we do, we may yet have a chance. And perhaps some of our sister species may have a chance as well. Cooperation is what leads to the success of multi-agency efforts. The most difficult part of trying to generate coherent, cohesive, concentric technical information about the ecology of the estuary is coordination between the various organizations and agencies who share responsibility for impacting or monitoring impacts to these underlying ecosystems. This applies to sample design, sample implementation, data analysis and publication, and presentation of results and their use. We in the Interagency Ecological Program are struggling with this point recently, even subjectively, as we have demonstrated oftentimes over the last 5 decades that we can be very good at just this thing. As examples, I'll mention the IEP Workshop, management analysis and synthesis teams, project work teams, the science management team, the IEP coordinators, and hundreds of co-produced peer-reviewed scientific publications, seminars, and trainings. Yet our collective governance, well, it sucks. Independent reviews by the Delta Independent Science Board, for example, say this, and they are not wrong. Somehow, we manage to get things done in spite of ourselves, and in spite of the fact that everything IEP always, forever, has been voluntarily agreed to. No coercion needed. Our cooperation just makes sense, particularly to those collecting the data and to those doing the analysis and creating the information. But sometimes as volunteers, we just can't seem to close the loop in ways that others might expect us to. Owing to the fact that we have at least 6 federal and 3 state agencies trying to coordinate legal, regional, state, and federal bureaucracies without the benefit of any central authority or even designated responsibilities, we've had considerable problems coordinating over the years. I'll not dwell on this point except to say that many potential approaches have been discussed. A few have actually been tried. I call to mind the GEI-led business practices reviews of the IEP in 2015, but none has completely worked. Perhaps now that we've been given the Delta Independent Science Board's review of our monitoring enterprise, it's been a couple of years now, there may be new arrangements to try. If there are any IEP agency directors that have any grand schemes of how to do this we haven't yet tried, we're all ears. This might be the most important point. People matter. The precise location of a scientist on an organizational chart in a particular state, federal, or non-governmental organization has some role to play in how we support and administer our science, sure, but passionate, capable, properly trained, and effectively motivated members of our science policy community matter much more than what position someone occupies. 1 or 2 influential and effective people can change the course of a project or program. Good ideas and effective implementation strategies can come from anyone, anywhere. It doesn't matter who you work for or what your rank is in that organization. If you have good ideas and are willing to work to get them implemented, then welcome on board. Let's hear from you. Traditional roles expected from within traditional organizational structures with traditional oversight and command and control have been an anathema to scientific creativity and successful project implementation during my time with the IEP. We need to find ways to identify and support good people and good employees, not overburden them with overhead and conflicting agency priorities. In particular, we need to help our managers and supervisors understand that a person's expertise and ability to work effectively in groups and across agencies is more important than whom they work for, what their job description reads at the time of hiring, or what their exact job title is. We need to find ways to cultivate and support successful employees and to prolong their retention in IEP roles and activities. Halfway, how am I doing? You all right? Number 6, science, IEP science should focus on ambient environmental context and action assessment. Applied science must focus on application and assessment of actions. Implementation and assessment of effectiveness is often missing from management relevant science actions. All the creativity and enthusiasm and research excellence in the world cannot stand up to our need for implementable consequences of esteemable science. I think, based upon my and many of my colleagues' experience, that this is the most difficult aspect of agency science to get right, the thing we've most struggled with, and the thing that policymakers and the public have and are demanding most emphatically now. Given all that we know, what should we do? It's hard stuff. Managing ecosystems is hard. Complicated relationships, constantly changing conditions, actors, forcing effects, things substituting in and out at irregular intervals. We're supposed to know what to do. Focusing our science on what is really important to the system might be a way to reframe our conversations in this regard. But we have to wade through a lot of, well, what do I want to do? And what do I think is important before we get to the crux of the issue, which is what is important to the organisms? What is beneficial to the system? Perhaps we can pursue a more estuary-oriented dialogue when thinking about what we should do. But who's negotiating for the estuary? It's not enough for your science to be cool. In the era, the era is past in the Delta and maybe in the whole estuary for it to be sufficient that you're doing cool stuff. Necessary for your individual mental health and motivation, perhaps, but not sufficient. The science we fund as an agency-centered community of scientists, as the IEP is, must meet the demands of agency managers, directors, and stakeholders. In particular, while the University of California is the research arm of the state of California, by definition, in my view, and can pursue interesting and cool topics from a majority intellectual and curiosity perspective, agency and departmental scientists have a mandate to get stuff done. We have to try to answer management policy priority questions and needs, as difficult as it has been for us to discover what those needs really are, or as tough as it has been to remain engaged with policymakers on what the ongoing science-based discussions are behind our problems, our challenges, and our victories. You may think your science infatuations are cool, but managers and agency leaders who pay for your time and do your science may not agree. In fact, it's probably an irritant to them that you cost so much. And you tell people how publicly thrilled you are to be doing cool work. I get that we want to share our enthusiasm for what we do. It's got to be more than cool. It's got to matter for the people who pay for it. This is related to my next point and bears some explanation to understand the subtleties of why. I'm not sure I have as much time as I would like to go into this, but even so, it really shouldn't just come down to money. And that leads me to this somewhat stark conclusion. Commoditization of science equals death by commission. To be successful, we must resist what I call the commoditization of our science. The people who finance what we do also need to meet us where we live and where we understand the world. We are human beings, and that a durable, credible, effective science is at base a very human enterprise. Getting together to discuss what ignites our passions and our curiosities at events like this one, the IEP Annual Workshop, is worth supporting, almost no matter the cost. Efficiency is a word that is often used in this regard, as in, we have to be more efficient with our science. But I think we all too often come to misuse the concept of efficiency, and we many times conflate more efficient with cheaper. The scientific enterprise is not best used to buy data that could be misconstrued into whatever information the purchaser needs to promote or defend from their particular position or perspective, or to determine some sort of predetermined practical outcome. Science is best used as a tool for the promotion of understanding, underpinning difficult conversations about policy choices, conservation options, the best way to model complexity, whatever. Science, in my view, is an extension of what it is to be human, to have a brain large enough to contain abstract thought and language, large enough to invent mythologies about our world, yet disciplined enough to know the difference between real and fake, between possible and probable, between likely and not worth worrying about. Communicating the essential implications of our science is really what we are and do. And it's not enough to buy a result or a preferred project alternative. We participate in knowledge generation, in whatever makes us wiser, not which generates more or robust or cheapest or better. What we do, what is it that we need to know to live better? Not what helps us get away with whatever it is we're getting away with. Also, deliberately narrowing our focus is not equivalent with being more efficient. Spending less isn't equal to being better. Some of what we need to know costs more than ever. That's a given. Then maybe we need to understand we can't have it all, and we must decide what we will have to live without. Our science and our understanding of what is most important to know is what is of value here, and we must have conversations about what is most valuable, not what is less expensive, and maybe not what is more efficient from a predominantly commodity-based perspective. A little bit of a corollary here. A lack of focus equals death by omission. Who chooses and how we choose our principal object of focus? IEP does not effectively do this well. Lacking a central clearinghouse dictatorship regarding strategic matters of direction for the existing monitoring enterprise and for the IEP generally, we tend to busy ourselves with project level tinkering and fine tuning rather than focusing on higher level issues of how best to deploy our limited resources in service of our higher and shared aims. Perhaps this is more of my own failing than that of our collective science enterprise, but the objective of convening a forum within the IEP where specific options and choices of larger direction setting discussions are had have been missing lately, and perhaps for a lot longer than just my term. Alternative venues for seeking such directional conversations such as CSAMP, CAMPS, DPICT, the Long-Term Operations Group, input from the Delta Independent Science Boards, recommendations from various reviews, it's all out there waiting for someone or something, some entity to pick it up and use it. I currently don't know where this level of decision-making and agency coordination lies. And finally, science matters less than we think. We have to be more than just scientists and understand that our science is not as important as we would like to believe. What leads me to say this? In a recent meeting of an advisory committee on which I serve, 17 senior scientists with something on the order of 300 combined years of professional experience in agencies, academia, national and international research and resource management programs were asked a simple question as they reflected on their experiences to help inform the organization we were advising. The question was, what is the one thing you wish you would have known at the beginning of your career that would have been of value as your career unfolded? 15 of the 17 science answered with some version of the following. Some of them holding their fingers up in front of their faces like this and saying, science is a very small part of policymaking. Think about that for a second. Senior members of our scientific community, our scientific and technical forebears, if you will, are telling us that all of our expertise, our schooling and master's degrees, our dissertations, research, PhDs, all our field work, time spent agonizing over the endless detail and learning of new analysis techniques and ways of communicating, what we found is only matters a little bit when it comes to policies that govern our resource use and management. That was pretty shocking to me. Maybe to you too. So, is it time to throw up our hands and go fishing? Well, maybe. But this advisory group that I was mentioning turned their attention immediately towards how we might best engage policymakers. That in addition to our hard-won technical competency and publication records, we need to focus on engaging most the people who choose not to be scientists or analysts or engineers. In other words, most people we deal with professionally are not scientists or engineers. They're policymakers. What matters to us is mostly unimportant in the broader world. Take that in for a second. Outside of me and my world, most people aren't interested in what I know. The same is likely true for you too. So what matters to policymakers and program managers? Well, we need to find out. And we need to discover how to talk with them and respond to them and to meet their needs. Otherwise, we're just whistling in the dark. So, in closing, I hope we have some time over the next 3 days to think about what our science should mean to our agency directors and our senior water managers as we celebrate how amazing and productive IEP scientists and project work teams are. Enjoy the workshop, take time to eat, sleep, and drink IEP for a few days. And I hope to hear from many of you as you create and support science dialogues as you make your way as agency, academic, consultant, or independent scientists over the next year. We need to keep talking. Ultimately, I think we scientists need to admit, and to ourselves mostly, that we are more human than we tend to allow ourselves to believe. While it can be motivating to believe that we are crusaders for the truth, I think it would be more effective if we understood ourselves as tools of the state or of the nation or of the people, if you will, and temper our expectations and ambitions accordingly. 30 years of service in and around the IEP and the San Francisco Estuary has shown me the power of our scientific investigative mindset, but it has also instilled in me a humility before nature and a very great caution about the likelihood of durable human achievement in the face of and surrounded by, in all ways, the nature on this planet. Thank you for all you do for the IEP.
Thank you, Steve. We'll now start in on our first session of the regular workshop on the physical and biological effects of flow. And to introduce the speakers, we have Sam Bashevkin.
All right. Thanks, everyone. Thanks for being here. So I'm Sam Bashevkin with the State Water Board. I'm happy to welcome you to our second session on flow. And without further ado, our first speaker is, is Brian Mahardja from the Bureau of Reclamation, talking about As the Smelt Swims.
All right. Thank you, Sam. Thank you all for coming. And for those of you who called in online, my name is Brian Mahardja. I'm with the Bureau of Reclamation, and I'm here to talk about a new analysis on an old topic, which is delta smelt spawning migration. Just for the handful of you who are not that familiar with the delta smelt basics, it's an endemic species to the estuary that is listed under both the federal and California Endangered Species Acts. It's an annual species, so only lives for a year. We're not sure what happens with the eggs as much, but we know that larval smelt spawns in fresh water in the kind of blue region there, roughly. In the summer and fall, they move towards the low salinity zone and the brackish water to rear. And when they get bigger in the winter, spring, they migrate back to the fresh water and starts the cycle all over again as they spawn. Just one wrinkle that's relatively new in recent years is that we start releasing hatchery adult smelt around this winter spring period during the spawning time. Okay, in terms of the spawning migration, when the fish move back to fresh water, it's triggered by this first flush event. So it's the first large storm of the water year that is associated with a turbidity pulse where the water can turn pretty chocolatey, as you see on the left picture there. And we know this because we start seeing a pulse of delta smelt in the past at the water export facilities in the delta when this first flash event happens. But last year in the smelt monitoring team that discussed kind of the water operations at the export facilities, there were a lot of discussion on this topic that kind of made me think there may be things to kind of look into some more on this spawning migration. There's talk about how once delta smelt move upstream, they don't really move anymore. And how when flows are high, maybe the distribution of the population move away from the influence of the water projects or the pumps. And there's even mentioned that maybe certain level of old middle river flow, which is controlled by export, there may be certain level that won't create any additional movement of delta smelt into the delta. So in light of that, a handful of us kind of want to tackle a couple of things. So we kind of want to better understand the spawning migration of wild delta smelt and whether or not it changes their distribution, as previously stated. And all we, now that we are really having mostly hatchery delta smelt in this winter spring period, we want to kind of understand whether the hatchery delta smelt behave similarly to their wild counterparts. Okay, so let's go on to the study objective one first. So the data set that we used here is the Delta Fish R package that's put together to combine all the fish survey data in the Bay Delta together. And we kind of looked at just the adult Delta smelt data, regardless of surveys. So the data primarily come from 3 surveys that are kind of listed there. So to kind of just gather just the adult Delta smelt data, I removed all the fish that are kind of below 50 millimeters in fork length. And keep in mind that I'm just using the November to April time period here. And then I summarize the data by month. In terms of the response variable, recall that I kind of aggregated the data set by month, looking at just captured Delta smelt data from the surveys. So at any month that had less than 20 fish, I kind of removed those data sets because I consider those as low sample size. And then that resulted in about 150, over 150 data points, monthly data points in total, representing tens of thousands of Delta smelt from all the way from 1976 to 2018. Okay, so what do I do with this Delta smelt data? First, we looked at the mean salinity that we see Delta smelt in parts per thousand. And the second part is the average least cost distance in kilometer between the Delta smelt catch data and the State Water Project facility or the Skinner Fish facility. And this is estimated in terms of as the smelt swims distance or as the fish swims rather than as the crow flies. So it's not Euclidean distance because we have this complex network of channel in the delta and fish don't fly. So just note that this is just, as estimated based on the monitoring efforts, right? So it's just whatever the data is showing us in the surveys. Okay, so in terms of predictor variables, there are a lot of things that we considered. The first thing is whether first flush has occurred. And the way we define that is just based on the regulatory document, which is looking at 3-day average flow at Freeport. We kind of threw in a lot of hydrological conditions. So all the flow variables that you see there, including turbidity as measured by Secchi depth in a couple of portions of the delta. And then we also looked at water operations, so export and OMR flow, and as well as temporal variables. One is month to kind of evaluate whether seasonality is a big component, and water year. So in terms of, so not water year type, but just water year in terms of kind of year to kind of capture any big events like the pelagic organism decline or the introduction of the invasive clams. And in terms of the actual model, we use regression tree, which is a nonlinear method of modeling that's fit primarily for predictive capability. So this is then advantageous in that it can handle a lot of correlation between predictor variables, because as you note in the previous slide, there are a lot of those variables that are collinear. And it's also helpful in that it's easy to interpret and maybe more useful for managers to kind of look at because they're going to understand it pretty easily. Okay, so jumping straight to the results. So this is the first response variable in terms of salinity that Delta smelt are in. So you can see the tree there. It's pretty straightforward. So, if first flush has occurred, you move to the left-hand side. If first flush has not occurred, you move to the right-hand side. In terms of the response or the predicted outcome, those are kind of the colored boxes at the bottom. So those are salinity in terms of parts per thousand. And then the percentage, it just kind of tells you the percentage of the observations in the data set that belong in those categories. Okay, so the first split is whether first flush has occurred or not. If it has not, then they occupy this low salinity kind of zone, which kind of makes sense. So 2.8 parts per thousand. When after first flush has occurred, they move into freshwater habitat as kind of predicted. But there's this determination. So if X2 has occurred, so it's dry conditions when X2 is high. When X2 is high, then they occupy 1.7 parts per thousand. But if X2 is low, so first flush occurred and conditions are pretty wet, then they occupy lower salinity at 0.5 parts per thousand. Okay, so the second part is we look at distance to State Water Project facility in terms of kilometer. And so this is kind of thinking about distribution rather than the salinity they occupy. Now the response variable is in terms of kilometer average. And again, the percentage is the percentage of the data set that belong into those categories. So note that the first node is about outflow. But this, just keep in mind that this may also partially represent first flush. So generally speaking, let me just move back real quick. So when the previous month's outflow is low, fish are actually further into the delta based on this data set, according to this data set. And then it looks at the X2 at 88 kilometers. So if X2 is high, that means it's dry conditions. They're actually even closer, so lower numbers into the state water projects. If you move to the right-hand side, so this is kind of farther away, so you see the colors are generally darker. It looks like the model is kind of picking up a sampling regime or maybe some kind of events throughout time. Because 1994 split is when Bay Study added new stations in the North Delta, and 2005 maybe capturing spring Kodiak trial initiation or the pelagic organism decline effect. It's unclear. But I think what's interesting here is that there's this last split on the bottom there, where it looks like Omar, which is influenced by the pumps. And this is just looking at years previous to 1994, where if Omar is really negative, more negative than 7,800 CFS, then they're really close to the facilities. And if the Omar is more positive, then they're farther away from the facilities. But I neglected to mention, just keep in mind that when the regression tree produced this output, we didn't pick the split. This is just based on the data set and where they see kind of like the best split for predictions. All right, so the key takeaway from this is that first flush always change the salinity that does not occupy, but not necessarily the distribution. That seems to be less determined by first flush itself. But generally speaking, so there are a lot of things that I'm not showing here. I cut down a lot of the analysis to get this talk down into 15 minutes. But generally speaking, distribution is actually closer to facilities before first flush, even though we don't see them at the facilities. But they're actually farther away typically after first flush, because first flush typically initiates the wet season. And then it's just interesting that the regression tree is able to capture changes in sampling effort. All right, so let's take a look at what's going on with the hatchery delta smelt. So the data set obviously is just whatever hatchery delta smelt we capture in the surveys after they're released into the delta. And because there's not a lot of recaptured delta smelt data, it's more of a qualitative evaluation. We first look at the swim path of fish, and then we're going to jump into the movement rate and salinity that the fish are in as it relates to first flush. Okay, so swim path, just keep in mind that because there was no unique data in water year 2022 when first experimental release happened, we're not really going to look at that because we don't know exactly where they come from, for swim path at least. Okay, so this is data from last year. This is the map where the blue square is where fish were released. The purple dots and circles are where fish were recaptured. And the larger the size of the circles, the more fish were captured at those locations. So recall last year was a wet year. And it's interesting that you can see that fish can move either downstream or upstream. They can move down to Suisun Bay, or they can move upstream to the export facilities. Or in some cases, so where you see the bottom kind of panels there, they can move up to the ship channel as well, despite high flow conditions. And another interesting thing to note is that this is looking at least cost distance, and really 3 Mile Slough represents kind of the shortest path and the most immediate path to the pumping facilities in the South Delta. So let's take a look at water year 2024, which I think is tracking to be above normal. In terms of water year type, it tells a pretty similar story that they can move upstream or downstream, but there seems to be more movement in this year for some reason towards Suisun Marsh. Okay, so let's look at movement rate. So the way we kind of measured this is we look at distance from release point in kilometers, and then we divide that by the number of days since release. I just want to note that it's going to asymptote at some point because after 30 to 50 days, it's just going to kind of become like 0 or 1 kilometers per day, just because fish don't travel infinitely. Delta smelt can't move to Shasta or the San Francisco Bay in this time period, right? So other things to kind of note in any case. So this is looking at time on the x-axis. And the shaded area is kind of conditions that are kind of denoted as first flush. Just notice that there are low recaptures and movement prior to first flush. But they've roughly, if we average them, they kind of average about 1.9 kilometers per day to 1.7. I just want to note that there was one fish that we observed that moved 23 kilometers in a single day, riding the kind of the flow there. Lastly, salinity occupied by hatchery delta smelt. The key takeaway is that most in the last couple of years, they remain in freshwater because they were released in freshwater, and a lot of them experienced first flush conditions almost immediately. But 2022, so the first year of release, it was a critically dry year, and the majority of fish never experienced first flush. Note also that water year 2022 was the only year when there was a release in kind of a brackish water area in Montezuma Slough and Suisun Marsh. So the takeaway from this is that hatchery delta smelt appear to remain in low salinity zone or maybe move towards low salinity when they never ever experience first flush. So maybe they were kind of confused of some sort, just like what we would expect from a wild smelt. Okay, so the key takeaways is that released hatchery delta smelt can either move upstream or downstream. In terms of movement rate, it's roughly about 2 kilometers per day, which is consistent with previous studies where they looked at kind of whole population movement in a different way. But there are pros and cons to either method. But just note that our study likely underestimates the true movement of hatchery delta smelt because of the fish doesn't move infinitely like I mentioned. And that hatchery delta smelt, just note that when they never experience first flush, they can probably remain in this kind of low salinity areas. All right, so that's all I got. I just want to thank all the past and current field crews that collected all this amazing data and the crew who put together the Delta Fish package and continue to update it this day.
All right, well, thank you. I don't think we have time for questions, but let me get the next one going. So next up, we have Marie Stillway Garcia, who will be talking about an evaluation of spring outflow to larval delta smelt.
Good morning. Thank you for that introduction and great talk, Brian. It's great to follow up to that. So I'll be discussing a project that we have been conducting. It's part of a multi-year project, but the results of which is also a multi-year project. So this morning, I'll be talking about the 3 years of larval delta smelt toxicity data that has a focus on the spring outflow. And this is going to be contributing to the years of 2021, 2022, and 2023. This follows 3 years of subadult delta smelt toxicity that had a fall outflow focus, which took place during 2017 to 2019. And during this time, I was able to get 1 year of subadult delta smelt with a focus in the summer. And I am interested in finding 2 more years of this data. So if anybody has any funding opportunities, please come see me at some point during this conference. So why spring? This is the conceptual model of delta smelt from Float Mass 2015. And so as Brian mentioned earlier in his talk, this is where delta smelt spawn in the fresh water. In terms of delta smelt themselves, the larval stage is the most sensitive stage. And there is some evidence outside of just the larval time point that delta smelt are in better histological condition during drought years as compared to wet years. And this is pointed out in Tay et al.'s 2020 report of delta smelt that's been caught in the wild over 7 years. In terms of the spring outflow, both the Central Valley and state water projects that has shown some evidence that can affect survival and growth of delta smelt. And spring outflow can also have a significant effect of salinity. In addition to that, spring outflow may reduce contaminants, but on the flip side, during a wet year, increased runoff may also increase contaminants. So we wanted to see what kind of contaminants were in the system during the time that we sampled. So with this in mind, our project goals were to identify toxic regions within the delta, characterize the types and the amounts of contaminants that are in the system, expose cultured larval delta smelt to these ambient waters in a lab setting where we determined effects on survival, condition factor, which is a measure of plumpness of the fish, RNA to DNA ratio, which is a measure of short-term growth, and histology, which is a biomarker of contamination. So our site locations, they did span both the freshwater and brackish water areas of the Delta, including the North Delta area that included the Tow Drain, the Deep Water Ship Channel, and Cache Slough. We got the Lower Sacramento River at Decker Island And then our brackish water sites were the Montezuma Slough and Grizzly Bay. So like I mentioned, these were cultured delta smelt. We conducted these toxicity testing on site at FCCL. And so because we had such a wide range of site locations and salinities, we included 2 controls with our toxicity studies. We had a regular freshwater control that we used FCCL culture water. And then we also included a high salinity control where we amended it with instant ocean to match the salinity of Grizzly Bay. But one of the really cool things about this particular project was that we used 1 cohort of fish to do all of our toxicity testing within a year. And so with each toxicity exposure test, as the fish age increased in this cohort, we sampled from that cohort to run our toxicity tests. And this is a little different than what we normally do in toxicity testing. You generally want to aim for the same age of fish and test it repeatedly so you can make comparisons over time. But because we had the first spawn date of the year, we were only able to use 1 cohort. But as it turned out, was really an interesting factor. And we were able to do this for all 3 years. So delta smelt at the larval stage is very small and they're very delicate, and delta smelt are very delicate in general if you've ever had a chance to work with them. And so we were able to design these specialty replicate buckets that allowed us to change the water without disturbing the fish. And on the picture on the right is an example of the larval fish at 55 days post-hatch. And this represented the size of the fish at our first toxicity exposure. And it's a little hard to see, so I've included a banana for scale. So in terms of our water years, like I mentioned, it's going to take between 2021, 2022, and these were both extremely dry years. And this was followed for water year 2023, which was a wet year. 2021 and 2022 were the second and third driest years in California on record. And in 2023, we actually saw one of the highest snowpacks on record. So this allowed us to see really big differences between water year and wet and drought years. This had a significant effect on site salinities. So these graphs you're going to see throughout the rest of the slides. Each site location has a different color. The freshwater control is in bright blue and the high salinity control is in red. And then our brackish water sites of Grizzly Bay and Montezuma are indicated by navy blue and brown. And in 2021 and 2022, we included a high salinity control to match these salinities. But in 2023, because of the high precipitation and outflow, actually all site salinities were within freshwater ranges, so we didn't include a high salinity control. The scale is in log, so to give you an idea of what these salinities are on the linear scale, there was huge differences between years and between sites. So what did the spring outflow affect our contaminant data? So you can see here with the sites, we saw a variety of fungicides that were used in agriculture, including things that are used on stone fruit trees, post-harvest treatment, a lot of stuff that's associated with the dormant spray. And we had a variety of contaminants that were seen in 2021, but we saw a significant reduction in contaminants in 2022. Now, these were both dry years, but like I mentioned, we were in the middle of a significant drought. And so 2022 was the third of that year. And so the difference in contaminant detections were really more related to the antecedent field conditions at that time. By 2022, this was the third year of the drought. And so we actually had a significant reduction in farmland in the Central Valley during 2022. Something like almost 700,000 acres of farmland were left fallow. Almost 300,000 of that was just rice alone. So it was really more related to the lack of agriculture taking place in the Central Valley rather than water year. In 2023, you would expect to see higher contaminants or maybe lower concentrations given it was a wet year. But you can see here from the data that everything kind of seemed to line up. So if you take a look at fluoridone, for instance, between the 2 dry years, we saw similar concentrations between the tow drain and cache slough, which are indicated by the green and the purple dots. But when you look at 2023, fluoridone was at a lower concentration for cache slough. But if you take a look at the fluoridone detections that were in other site locations, those closely match up with the concentrations that we saw in 2023. Meanwhile, taking a look at hexazinone, again, we saw similar concentrations. We did have a couple instances of higher concentrations in 2022, and especially if you look at other sites not related to the control. And in 2023, we saw, again, lower concentrations. This isn't exactly what we expected, so this is pretty interesting. In terms of the trace metals, concentrations and detection frequencies were similar across years. Really, the only difference was that we saw a higher frequency of detection for mercury in 2022 compared to the other 2 years. Now, in terms of results, I'm just going to give an overview of the annual differences we saw. I have too much data to really go into inter-year or site locations throughout the years. And so each year is going to be bracketed with our statistical analyses. We either ran ANOVA's or Kruskal-Wallis test, depending on the endpoint, and different letters are going to indicate different statistical differences. So what we saw in survival was that Delta Smelt in 2023 had better survival compared to 2021, and the fish in 2021 had better survival than those in 2022. And you'll notice in the 2 dry years of 21 and 22, we saw high survival in the high salinity sites, including the control, and all the freshwater sites had significantly reduced survival in comparison. So condition factor, again, this is going to be a measure of the plumpness of the fish. And we saw the same pattern in condition factor as we did in survival. So fish in 2023 were in better condition compared to 2021, and those fish were better than 2022. However, when you look at the RNA DNA ratio, we saw the opposite. So fish in 2022 actually had higher RNA to DNA ratio compared to both 21 and 23. And I'm not entirely sure why this is. We were able to use the same cohort of fish and they did have similar hatch dates, but the fish that were used in 2022 were a little younger than those in 21 and 23. The dates ranging anywhere between like 3 and 6 days difference. And so since younger stage fish do have a higher rate of growth, it could be contributing to the differences we saw during this year. Now, in terms of histology, we looked at liver and gill lesions, and this is an indication of contaminant exposure. And so on the left y-axis, you're going to have the lesion score, and the higher the lesion score indicates the higher degree of damage that we saw. And lesion scores are really an aggregate of the number and the types of lesions that we saw, indicating the type of damage. And so during our wet year, during everything freshwater salinities, we did see significantly more liver lesions than we did in both 2021 and 2022. And liver lesions were essentially absent in 2021. But in 2022, we did see some mild lesions in the fish that were exposed to Cache Slough and Decker Island. And then we also saw the addition of fish exhibiting liver lesions in the toad drain. Now looking at gill lesions, we did see differences in gill lesions across years. Again, fish in 2023 did have a higher prevalence and higher lesion scores compared to both 21 and 2022. But you'll see here that across the years, regardless of dry or wet years, that we did see a higher lesion score in Delta Smelt that were exposed to Cache Slough, both in 2021, 2022, and again in 2023. To give you an idea of the lesion scores that we're seeing, this is a picture of a gill that was from a Delta Smelt that was tested in 2021 that was exposed to water in Cache Slough, and this was during our last toxicity exposure that took place in May. And so here you can see that both the primary and secondary lamella have fused together, which is going to result in reduced gas exchange for the fish and make it hard to survive. And to give you an example of what a healthy gill looks like, we've got the primary lamella that are separated as well as the secondary lamella that are also separated. And so you can see here by the fish that has the lesions in the square that those are all fused together, and that's going to be detrimental to the health of this fish. So with that wrap up, our conclusions, I didn't touch on it too much, but we did see a westward movement of X2 due to high outflow. And this was seen by our freshwater salinities of our brackish water sites. And this isn't necessarily a surprise, but it is interesting to see exactly how far X2 moved westward. In terms of the idea that Delta Smelt are in better histological condition in drought years compared to wet, in our particular study, it turned out that the weak fish died. And so really all you saw were healthy fish remaining, and those were in good histological condition. In comparison, during the survival in the wet year, we saw better survival overall, but those fish also had a higher prevalence of lesions. And so you're going to really have to determine what is better. Is it better to have more fish that might have damage or less very robust fish? It's kind of difficult to say, and it's probably worth looking into further. As we saw during this year, and I've seen in other years as well in other studies, Cache Slough does tend to be problematic for Delta Smelt. We saw this in survival and histological condition as well. And then again, with the chemical analyses, we saw that antecedent field conditions influence contaminant detections, but not necessarily concentrations. And this kind of went against what I initially thought would happen with this data set. So I'd like to thank all of our collaborators, without which we weren't able to make this project happen. Of course, all the other years as well. And so it was a great team effort by everybody. These are just a little bit of citations of my fun facts that I gave throughout my talk. And if I have time, I'm happy to take any questions.
I think we have time for probably one question. We've
So you saw higher lesion scores in cash slough and I think the toe drain also in 2023, correct?
Yes.
Do you have a sense for the relative influence of higher flow conditions versus the application of pesticides or other contaminants in that area in 2023?
That's a good question. Yes. So I should probably note that we didn't test for every single contaminant class there was. And so I may be missing contaminants that may have been in the system that our detections didn't see. And unfortunately, the pesticide use data isn't released yet for 2023. So I can't say for sure. My guess would be because the water year was wet, there might have been an increase in agriculture that would be related to an increased use of pesticides and herbicides. But again, I can't confirm that. But given patterns I've seen in previous years, it wouldn't be too far off to expect that type of thing to happen.
Okay, thanks.
great. Well, thanks a lot.
All right, thank you.
Next up, we have Paul Stumpner from USGS, who'll be talking about X2 and the salt field dynamics.
Thanks, Dan. Hello. As Sam said, Paul Stumpner with the USGS. This study is a collaborative project we worked on with the state board, and I'll be going over about 2 years of data we've collected so far and it's improving our understanding of salt field dynamics in the estuary. Specifically, we are looking at ways in which the broad scale salt field movement of X2 can have effects on the ecosystem. So first off, I just want to go over a few conceptual models about salt movement in the estuary. This figure shows a longitudinal cross section of the North Bay from San Pablo Bay through Carquinez Strait into Sassoon Bay and then the Sacramento River. And up here shows the longitudinal salinity profile. The steeper the slope indicates stronger salinity gradient, which is important for salt field movement because you get these cells of gravitational circulation or two-layer flow. And that can be stronger in these deeper channels as well, where the salinity gradient's higher. And gravitational circulation is the mechanism that creates stratification in this estuary and in other estuaries as well. And this slide shows a high flow condition, and we can contrast that with a low flow condition where the slope of this line is shallower or not as steep. So therefore, the salinity gradient is weaker. That results in weaker gravitational circulation and ultimately lower values of stratification. To go a little more in detail about gravitational circulation, this is two-layer flow that results when fresh water flows out of the system and replaces the saltier, denser water that then has to flow back upstream. And this creates two-layer flow as indicated by these yellow flow arrows. What also happens at kind of the convergence of the freshwater saltwater interface is you tend to accumulate material, whether that be detritus, sediment, or phytoplankton is indicated by diatoms in this schematic. So what we're primarily looking at for this study is the effects of stratification and what effects they might have on the ecosystem and specifically phytoplankton blooms. So on this figure on the left shows unstratified water column where it's well mixed from top to bottom. So phytoplankton that can grow in the shallow photic zone can also be mixed and transported down to the benthos where they're exposed to clams, which are a big loss term in this equation. When you contrast that with a stratified water column, there's two layers of fresh water and then saltier water. And those two layers don't mix well. So therefore, phytoplankton that grows in the upper water column is not exposed as much to the benthos and grazing by the clams. And they have more time to spend in the photic zone where you can increase the growth. So the main things we're looking at for this talk and for this study are what are the timescales and variability of gravitational circulation and stratification? And are these timescales of ecological relevance, both to tridal and phytoplankton production? And as I mentioned, I'm going to be focusing more on the phytoplankton production part of this. So our high resolution monitoring included installation of new monitoring sites shown by these orange dots. We're monitoring bottom salinity. So this gives us a better spatial resolution that's available in contrast to the green squares that show the surface salinity measurements that have been traditionally been used to estimate X2 and to understand the salt field. But for this talk, I'm going to focus primarily on data collected in this region, specifically channel marker 24, includes all our physical data, velocity, stage, et cetera, and salinity. And then we had a few sites that I'll show later that has some chlorophyll data. So the high level take home points for our salinity monitoring is that stratification is more frequent and of higher magnitude than has previously been observed. The frequency and magnitude of stratification also varies, has a fortnightly, a monthly, and seasonal component to it. And these timescales of variability are primarily due to the phasing of the spring neap cycle and the diurnal inequality of the tides, which I will talk about briefly in a little bit, or probably next slide. And based on our observation of stratified conditions, these can occur for the course of several days to 5 days. And this could enhance primary production if it co-occurs during daylight hours. Since these timescales are in the order of the timescales it takes for phytoplankton to show significant growth. So we developed a couple of metrics to assess the phasing of stratification in daylight. One is called the ebb flood amplitude. And this is done to identify periods of weak tides and weak mixing when stratification is more likely. The second one is called potential energy. And this is simply solar radiation that's integrated over the stratified periods. And this is used to assess the likelihood that physical conditions exist for increased primary production. And I'll note that this is, we think this is a necessary, but not necessarily, what's the right word, sufficient metric to allow phytoplankton production because there are other variables that need to be considered. So if we look at a water level data here, I'll briefly describe what I mean by diurnal inequality. And this is simply the difference in either two ebb or consecutive ebb or consecutive flood tides. So in this graphic, you can see these two consecutive ebb tides are quite different. That means the first ebb's quite a bit higher than the second ebb, so you get higher diurnal inequality. Contrast to another time period where the two ebbs are very similar, and this is lower diurnal inequality. And we see during these periods of higher diurnal inequality, we have weaker tidal currents because the change in water level is analogous to the speed of the tidal currents, or is correlated, I should say. This figure shows water level on the top, velocity and vertical velocity, which is a surrogate for mixing, and then salinity on the bottom. As you can see, these periods of weak tides equate to periods of weak currents and weak mixing, and that co-occurs with these stratified periods. If we look at a longer month-long time series, we use these envelopes of the water level to distinguish weak versus strong tides. And we can see that these lows in this metric, the ebb flood amplitude, correspond with periods of increased stratification. And you can see this has a fortnightly cycle, about 14 days apart. And you generally get these periods of 4 to 5 days of stratified conditions. It's not continuously stratified, but it's stratified for most of this time period. The potential energy metric we used, water level is on the top plot. Stratification is on the second plot. These gray periods indicate the time period of stratification. The third plot is solar radiation. So you can see on the plot on the left that we have many periods of overlapping daylight and stratification. If we simply integrate the solar radiation over those time periods, that's indicated by this orange dot here. So the closer that is to the total energy or the integrated radiation over that day, the more stratification is occurring during the daylight hours. So in this plot on the left, stratification and sunlight are in phase, whereas this plot on the right, stratification and sunlight are not in phase. So this has this cycles throughout the year. So if we look at longer time series, we have the ebb flood metric on this second plot. You can see that variability throughout. This is about 1.5 years. So you can see how the weak tides vary throughout the year and fortnightly and monthly time scales. You can also see that the weak tides are generally correlated with these higher stratification periods shown in this third panel. I should know there are some time periods where the weak tides aren't a good indication of whether we're going to have stratification. That's due to other, physical parameters such as wind mixing that needs to be considered. What we also see is that the consecutive days of potential energy or the phasing of stratification in daylight is typically higher in the spring or early summer. And that has to do with how the phasing of the tides and the sunlight comes in and out of phase throughout the year. So what effect does this have, if any, on phytoplankton? And we're using chlorophyll A here as a surrogate for phytoplankton. So we have water level data on the top, this ebb flood amplitude metric on the second graph, stratification on the third panel, and then that potential energy metric on the bottom panel. And then we have time series of chlorophyll A near our sampling location, near that channel marker 24. And we can see during these periods of higher diurnal inequality is related to these lows and the ebb flood amplitude, which is also corresponds to higher stratification and more consecutive days of stratification in daylight. And then that also is shown to be correlated with higher levels of chlorophyll as well. So what we can say based on the results of this study is that stratification during daylight hours is more frequent and of higher magnitude in the spring and early summer. There is some evidence that stratified conditions in Suisun Bay area can support higher levels of phytoplankton. But there are other controls on phytoplankton that we weren't considering in this analysis. But these periods of weak tides and by extension stratification can be predicted. So that way we can design field studies to study the role of stratification on phytoplankton production. With that, I'd like to acknowledge our funding source, the State Water Board, and then just all the great field personnel to make this data possible and these results possible. So thank you.
We have time for a quick question if anyone has one. We wait for the microphone.
I was wondering how you thought attenuation would interact with some of the, potential energy, dynamics you would observe.
Light attenuation?
Yeah, light attenuation.
Yes, I thought about that, but haven't analyzed that. Turbidity is a pretty good surrogate for light attenuation, and that definitely has seasonal variability. So it tends to be higher when you get flushing of the water. But there's also tidal variability, and it's generally lower, higher up in the water column during these stratified periods, and higher in the lower portion of the water column. Right, thank you.
Thank you. All right, so next up, this last talk, just for the future audience watching the recording, this talk will not be recorded, but the next talk is from Brian Healy talking about management analysis of Sacramento River fishes. All right, well, thanks everyone. That is the end of this session. I believe there's a 20-minute break after this until 10:20 and then the poster session, but anything else?
Yeah, I might as well give you your instructions for the poster session because you're not going to be coming back here. One of the feedbacks from last year's workshop was that you wanted more time for posters. So we have a 20-minute break and then you have a 2-hour poster session. So that's 2 hours and 20 minutes that you can do with what you will in terms of posters. And it rolls right into lunch. So you don't need to be back here until 1:55. So the posters are upstairs and to the left at the end of the hall. You should see signs, really great group posters. Should be a lot of fun. That officially goes through from now until about 12:25. Lunch is 12:25 through 1:55. If you have signed up for the mentorship lunch, please meet at about 12:55 at the top of the stairs. You do need to be let into the room that luncheon takes place in. So yeah, meet at the top of the stairs. If you brought your lunch, feel free and find a spot around the ground floor here. Not in the auditorium though, to eat. If you'd like to explore a little bit on the workshop website, you can access via the QR code. There's a little area guide that has some recommendations for local restaurants, but just be back here at 1:55. And yeah, enjoy the poster session and your break. Again, there's coffee in the lobby that should be drunk in the lobby. And thank you to ICF for providing that.
And if you haven't already sent up your slides, please come meet me right away. Thanks.
Take your seats and settle down. Our next session is chaired by Mr. Daniel Ellis from CDFW, and it is called Reconnecting Delta Food Webs.
If everybody as they're filing in can just be a little bit quiet, we're going to get started. So as Rosie said, this session is called Reconnecting Delta Food Webs. As probably all of you know, we have a very small amount of wetland remaining in the Delta, and many of these talks are going to discuss how we can reconnect those wetlands with the adjacent channels. So our first speaker is Lillian McCormick, who is a postdoc with the Delta Independent Science Board, as well as Bob Naiman, who also has an affiliation with the University of Washington. Slideshow.
Okay.
Yeah, there's,
Oh,
there you go.
awesome. Thanks, Daniel. Hi, everyone. Thanks for joining us right after lunch. I know this is a tough slot because this is when you immediately start to feel sleepy. So I will try to be more animated so that you don't fall asleep. So thanks again, and thank you again to the organizers for setting up this workshop. It's a really cool way to connect with the community. So this talk will be slightly different than some of the other talks in the food web section. We're talking about towards integrated food webs, understanding the benefits and science needs for incorporating food web interactions into management in the Bay Delta estuary. So first, I'll start off with an introduction. So for the last 2 years, I was a postdoctoral researcher with the Delta Independent Science Board, but what actually is the Delta Independent Science Board? And so the Delta Reform Act of 2009 mandated the Delta Independent Science Board, or Delta ISB, to provide oversight of scientific research, monitoring, and assessment programs that support adaptive management in the Delta through periodic reviews. So typically in these reviews, the ISB evaluates the state of science in general and assesses the adequacy of these programs in the Delta to address management issues. However, they do not make, recommend, or endorse policy decisions. So why study food webs? This is obviously not a new topic in the Delta, but it's a really important one and close to my heart. So the ISB review actually focused on upper trophic level food webs. And the objective of this project was to assess the state of the science related to understanding these upper trophic level food webs in the Delta that could benefit individual species and ecosystem management. So this really complements some of the existing work that's been done on the Delta on lower trophic levels. So we really wanted to focus on food webs because changes to one species, as I'm sure you all are aware, can affect the abundances of other species, and food web interactions can play an important role in many issues across the Delta, including habitat restoration, native species, non-native species, climate change, flows, and nutrient or contaminant cycling. So the IFB conducts these thematic reviews. So these have a kind of long process that they get started with. So first, there is a literature review. So essentially learning what is known about food webs in the Delta, which sounds fairly easy, but let me tell you, it is a huge block of literature to go through, as you all are probably aware. They then create a document called the prospectus, which is generally a document that describes review planning and also receives public feedback to kind of direct the review. We then conducted a series of what we call Delta discussions, where we talked with the community and really tried to answer a couple of different questions. So first, how can knowledge of food web interactions really be used to benefit the management and science priorities in the Delta? And then also, we really wanted to understand what themes, questions, or products in a workshop that would be helpful to the community. So that leads me to the next bullet point, which is that we then created this 2-day public workshop where we talked about a wide variety of different food web topics. And we really tried to understand how a better kind of knowledge of food web dynamics in the Delta could contribute or improve management, as well as learning about new tools and new methods that could be needed to incorporate food webs into the existing monitoring programs and into Delta management. We then published are working on publishing a written review that summarizes the key findings and some recommendations. So that's now in its public feedback stage. So I encourage you all, I'll get to that later in the presentation, but I encourage you all to take a look at that. And the areas that are highlighted in green are areas where we had public participation or feedback. So I'll jump right into the findings. So the Delta discussions were a really, really cool aspect of this project for me in particular. So this is really where we came to the community and tried to learn as much about what was important to the community with respect to food webs as possible. So really from these discussions, we came up with kind of 4 main components of topics that related to food webs. So this is structure and processes, so how altered landscape or food availability or ecosystem complexity affects the system. Human influences like modified flow regimes, contaminants, or harmful algal blooms. Different tools and methods that could be used to understand food webs and understand food web dynamics as well as modeling approaches. And then once you have those data, what can you do with them? And continuing to kind of monitor these data sets, make sure they're publicly available and usable, and also make sure that the synthesis is being done about this kind of wide amount of data that the system has. Okay, so as I mentioned, obviously this system has a ton of science that has been done with respect to food webs. It's a well-monitored, well-studied system. But so really when I say science gaps, I mean things that could be integrated into our existing knowledge better. And so these were some of the things that were highlighted in Delta's discussions and also in the Food Webs workshop that could use some additional work. And I'm sure many of you in this workshop will demonstrate areas where this research is happening. So we said that further examining the roles of detritus in system productivity and understanding the flow of energy through food webs. And this kind of builds off work that was recently funded by the Delta Science Program on kind of detrital energy movement through the food web. Understanding the bioavailability and linkages of primary production to zooplankton and then subsequent trophic levels, especially with respect to the spatiotemporal heterogeneity of the system. Better characterizing the role that benthic invertebrates play in the food web, and this is both as primary or secondary food sources or alternate food sources, and also with respect to competition with other species. Quantitatively understanding the role of the most abundant fish species in Delta food webs. Not surprisingly, the regulatory agencies kind of require us to focus on, three main species in this system. So really understanding the most abundant species rather than just the listed species may be important for understanding food web dynamics. Also understanding the flow and consequences of contaminants throughout the food web and the ecosystem, and evaluating the role of predatory birds and mammals with respect to the food web, such as the sea lion shown in this image. Okay, so the review goes through suggests four main applications of food web science that would be appropriate for Delta management. And I'm just going to give one as an example today, but if you go look at the review, there are additional details in that report. So I'm going to talk a little bit about ecosystem-based management and how understanding food webs could contribute to efficient ecosystem-based management. So we give an example management question. So for example, how do changes in environmental conditions affect food web interactions? And so really what's cool about understanding food web interactions is that you can then start understanding how changing conditions will indicate differential vulnerability to those changing variables, or the differential vulnerability of species to environmental changes, because some of those key ecological species may not just be the listed species. So ways to kind of move forward with this would be to connect laboratory or field experiments to evaluate sublethal effects of stressors on species into models, or developing some of those models or a series of models that would be designed to predict changes in species interactions over time. Okay, so trying to move fast through these. I'm going to briefly cover the recommendations. Again, these are just, this is just a brief overview as much as I can do in a short talk, but please do see the report for additional details. Okay, so a meaningful application and continuous evolution of understanding food web processes and the resulting modeling applications for the Delta really requires a focused interdisciplinary collaboration among agencies, universities, the public, indigenous tribes, and other interested parties. And this is largely because this process and food web dynamics span kind of the mandated regulatory area of multiple agencies. And so it's really important to have this be kind of as large of a collaboratory as possible. So the idea of a kind of focused collaboratory is not a new suggestion in the Delta, and I want to recognize that there has been work with the integrated modeling framework strategy from the Delta Science Program, as well as the idea of a collaboratory was suggested in the Science Needs Assessment and Workshop from 2020. And there are examples that we can pull on from other areas. For example, the Chesapeake Bay, the newly formed Chesapeake Global Collaboratory, which you can see a diagram from in the bottom of this slide. So participants in this collaboratory would decide on key management needs and science questions, and where possible, which some of this is already occurring in the Delta, but activities identified the plan would be established as testable experiments, built with hypotheses built in and be able to be tested. And so the ISB, I think, thinks that this could be a really effective way to test the use of a collaboratory with respect to Delta management. So this collaboratory would really be narrowly focused on Delta food webs, which I know is not a small topic, but they would really focus down on Delta food webs, and that could be an effective use to see how collaboratories might work in the system. Okay, number 2 would be to adapt Delta research and monitoring programs to explore key aspects of food webs. And this would not surprisingly rely on lots of collaboration and the best available tools and methods in the system. Ideally, these would all focus on science gaps and connections to existing knowledge that we have in the Delta. And this is also a plug for saying that food web relevant data need to be regularly updated, quality controlled, and made accessible. And this was something that we heard multiple times throughout the workshop. Number 3 is to employ appropriately scaled food web models as determined by management questions and environmental driving forces. So when I say appropriately scaled here, I mean that they incorporate the relevant processes and trophic interactions to learn relationships between environmental conditions and also the species of interest. And so this includes temporal and spatial scales, as well as the level of complexity. So for example, if you look on this slide, both of these are models showing food web dynamics. But these, while they all show, they both show food web interactions, they differ and they can be built off of hypotheses and be tested. They differ in their level of complexity and their spatial temporal scales, and therefore their utility for being used to understand different science or management questions. So picking the model that's appropriate for your management questions is really, really important. And then finally, it wouldn't be a Delta talk if I didn't have an adaptive management slide. But really, I think that this could be a very, very useful process where linking food web models to management questions and actions, monitoring and also empirical studies could be used, could use an adaptive framework. And that way you would have set decision triggers to figure out when you should adapt the monitoring program or change your food web model. Okay, so briefly conclude, and then I'll turn it over to Bob for some gems of wisdom on the way forward. Advancing food web science in the Delta will serve a broad range of management applications. Collaboration and adaptive management will increase effectiveness and efficiency, which, as Steve reminded us, does not necessarily mean lower cost. And it should improve capacity to forecast the effects of management actions and climate change on fish and other aquatic species. So with that, I'll turn it over to Bob to talk about the way forward.
Okay, great. Thanks, Lillian. Well, this is probably the easiest talk I've given in years. All I have to do is let you know that this is an ambitious undertaking, as you might imagine. It will take probably many, many years, if not a decade or more, for it to happen, but it'll only happen if you as a science community and a broader community really embrace the idea of using food webs to look at processes within the Delta as an ecosystem. And along those lines, it's going to take a number of follow-up workshops to get there. And we hope that these will happen over the next 2 or 3 years. But again, if you have ideas you would like to convey on how to move forward with various workshops or topics and so forth, then please let us know. It's a whole community effort. It's not just the Delta Independent Science Board, trying to push an idea upon everybody. It's something that if you like it, embrace it, and we'll go forward. Now, would like you to take a look at the report. It's not long. The executive summary is only about 8 pages. But it's open for public comment now until June 7th. And if you have any comments at all, please send them in and we will make sure that we respond to each and every one of them. So thank you.
Our next speaker is Emily Richardson, who focuses on phytoplankton, nutrients, and water quality.
This is Hi, I'm Emily Richardson. I'm a researcher with the USGS California Water Science Center's Biogeochemistry group, and I have a problem. I have too much data. I have too much data across the bay, into the delta, and particularly within the cache slough complex. Now, I realize that this is the best problem a person could have, but for anyone who has tried to analyze big environmental data sets, you know that there are many hurdles to jump over. So I have stumbled into a way to process, visualize, and analyze too much environmental data that I'm going to be sharing within the context of the cache slough complex. Let's begin. The cache slough complex is located in the North Delta. Zooming in, we can see that, as the name implies, it is a complex of water bodies surrounding the cache slough, including many different sloughs, lots of human-made and altered waterways, and 2 partially submerged islands. Now, in terms of water quality and water age, we see very strong gradients from north to south. This is due to a few reasons, including agricultural diversions to the north and tidal influence to the south, pumping in fresh Sacramento River water. And with every area, we see seasonal and climatic shifts, but unique to the cache slough complex, in dry years, we see high turbidity. Now, when we think about turbidity and when we think about the delta, we think Sisseton Bay. But in very dry years, the cache slough complex can actually become the most turbid location within the delta. About our too much data. Our group specializes in high resolution water quality mapping, which means we kit out this zippy boat with 2 fixed step pickup tubes strung up to a string of a ton of different instruments, and see Katie O'Donnell's poster for a better explanation. But what this allows us to do is to collect data continuously from a moving boat. And every single second we collect a data point, and that allows us to collect very high resolution water quality maps. The advantages are that this technique is unmatched in spatial resolution. We are able to very finely detect steep gradients and patches, which you cannot do with traditional discrete sampling. And it really unlocks an unlimited possibility for data exploration like I will be showcasing here. The one disadvantage is that it is unfeasible to conduct very frequently at high temporal scales. And that leads me to one of my problems. So you can see here, we have conducted these surveys in this highlighted location of the cache slough complex since 2015. Now you'll see that the years, the months of the surveys are not equidistant in time. And even within survey, there are differences in the amount of data we get. So to try to mitigate some of this, I've broken up my data into wet season and dry season of each water year. Now, this doesn't solve every single problem. Notably, there was no data collected in the wet season of water year 2020. So there's a bit of interpolation that'll have to happen, but it's the best I can do and I'm gonna roll with it. Okay, so when we go out on these surveys, we stop routinely at different stations to collect discrete data. So this also creates a bias towards more data in those specific locations. So to help mitigate that, what I have done is sliced my study area into 0.1 mile polygons. And for any one of those little polygons, for any one of those defined time slices, if I'm missing data, I'm going to interpolate. I'm going to use k nearest neighbors with a small time window and a small space window to try to find data and take the median of any data I can find and assign it to that missing data. And then once I have my larger data set, I can now start to aggregate. I will take the median from each of these polygons, from each of those time points. And I do this for all of my parameters, but the ones I'll be showcasing here today are water temperature, turbidity, dissolved inorganic nitrogen, and chlorophyll fluorescence. But still have a problem. If I'm trying to analyze time series data and I'm looking at a map, I can't do that. I cannot use a 2D map to look through time. So we're going to add time as a dimension. So what you see here is time going incrementally up from the bottom up. So the time slice furthest to the bottom is my farthest away record, wet season, dry season, chronologically until you get to the very top layer, which is my most recent data set. I can then plug in those median calculations into each of those cubes that I've created. And we can start to visually explore the data. So here's water temperature, for instance, and you'll see here that the lighter colors are the lower temperatures and the darker colors are the warmer temperatures. So you can see very stark striations in the data, which is good. This means we are doing something right because effectively by binning by wet season and dry season, we are binning by cool temperatures and warm temperatures. We can do stats with these data. We can test every single one of those cubes individually across its own time slice and within its own position across time. We can calculate the z-scores for all of these cubes, combine them and get an entire cash-slew complex trend. So the trend for temperature is that there's no trend. There's no significant difference in water temperature through time with these data. I can also look at it a different way. I can apply a hotspot analysis, which also looks at every single time cube, and it looks in a more conservative way. It'll average 2 cubes up and down, which is a year out, and 4 spaces to the sides. So we're going to take that average, this rolling average for each of these cubes. We're going to compare each of those for the average of the entire data set. So now we can see what areas are statistically higher and what areas are statistically lower. So temperature again, the red colors are the statistically higher data. The blue colors are the statistically lower data, so cooler temperatures. And then the gray areas are not significantly different. So a few things we see here are that water year, the past few years have been pretty warm, and we can see that within our data. We can also see a blue stripe down the middle, and that's associated with water year 19, which was one of the wettest on records for this data set. Let's look at some other parameters. So here's turbidity. We can see that turbidity is significantly decreasing in the cash-slew complex according to these data. Now, remember when I said that during very dry years, the cash-slew complex is very turbid, and the past few years have been pretty dry? Well, there seems to be some sort of issue here, and what I'm gleaning from this is that the cash-slew complex is following the delta-wide trend of decreasing turbidity. Dissolved inorganic nitrogen. This is an important metric because it's the sum of ammonium and nitrate, which are the 2 forms of nitrogen that are most readily available for phytoplankton to uptake as a nutrient. And my disclaimer on the data here is that we did not start collecting continuous ammonium data until 2017, so there's a shorter time frame showing here. And I also didn't have enough data in Lindsay Slew to comfortably interpolate, so I sliced it away from the data set. All right, a few things we can see here. We can see those clear gradients from north to south in terms of dissolved inorganic nitrogen. And we can also see that dissolved inorganic nitrogen is statistically decreasing as well in the cash slough complex. Now, not entirely shocking because the Regional Sand Wastewater Treatment Plant implemented a biological nutrient reduction process that significantly decreased the nitrogen in their effluents. So we can see that potentially the impacts reached to the base of the cash slough complex, but we can also see in the northern portion of the cash slough complex, it was always a cold spot. So it does not appear that that effluent ever reached the northern portion of the cash slough complex. My favorite, chlorophyll. We can see chlorophyll also significantly decreasing in the cash slough complex. So every spot that was a hot spot is now not significantly different. Every spot that was not significantly different is now a cold spot. So the takeaways are that nitrogen, turbidity, and chlorophyll are all decreasing. So at this point, I wish that I had that Taylor Swift pop-up. I'm the problem. It's me. I'm the cash slough complex. And also, space-time cubes are very fun and very informative, which is a winning combination. And also incredibly important is that we cannot perform these surveys without funding, without our funders. So thank you to the many and counting groups that believe in this work. We thank you. And also with too much data comes a whole heck of a lot of people to plan for, prep for, collect, process, analyze, store, publish, you name it, we've got a person for it. So thank you to our amazing group. And I'm going to leave you on a bit of a cliffhanger. Some of the parameters that I've analyzed but didn't have time to go over. And I think I have time for questions. Yeah, okay.
So for those who are going to be doing questions, just make sure you speak into the mic.
Okay. You group the cold and warm seasons, but with the weather we've been having, the cool seasons have been pretty warm in the years when they're dry. And we've had some remarkably cold years recently when they've been wet, but that doesn't seem to be that distinction seems to not hold up as well as it used to. Do you see that showing up in your analysis that things have changed through your time, and that the wet seasons are warming up sometimes?
Let's go back to water. So I would say that the top slice here is the dry season of water year 2023, which was wet. And it's also very warm. So potentially we are seeing differences.
Look, that it's just it's nice to think about wet season and dry season because that is kind of what we live by.
Mm-hmm.
But it does seem like they're changing too. So yeah, this would be a question I would ask of your modeling. How much is that changing? Where is it changing? All that.
Well, I think that's the good reason to do the hotspot analysis is because it's not taking the whole average. You can look at every individual location and see how it's different through time and through space. So I think that this tool is the one to use if you're trying to pick out if there are going to be changes through time that might be here to stay.
And how do you choose what you're going to look at? With 16,000 questions you could ask, how do you choose which ones are worth your effort?
Well, my data is primarily water quality and phytoplankton. That's my bread and butter. So those are currently the only ones I'm looking at, but I do have plans to bring in long-term monitoring data for zooplankton, phytoplankton, clams. We have just also the unlimited amount of data since we live in an open access society. I can do this with anything I want in any location I want. So it's really, maybe we'll check out the ISB document and the science action agenda and go from there.
Okay, we're out of time. Okay. So thank you, Emily. 1 second. Okay, our next speaker is Kai Ross.
Hello, everybody. My name is Kai Ross. As you just mentioned, I'm a senior biometrician from Kramer Fish Sciences, and I'm here today to talk to you about continuous zooplankton observation, detection, and enumeration. And really couldn't have gotten a better slot to talk about this as following that continuous data presentation. So that's going to save me a little bit of time. Maybe we'll have some time for QA. So first of all, zooplankton are important. They're generally speaking the second trophic level. They're combined, linking those primary producers into the rest of the food web, which, as you guys are well aware of, includes many species of concern. And they play a direct role in both fish recruitment and overall biomass. So they're definitely an important player in the world of food webs. That being said, the majority and previous sampling techniques are often discrete samples. They're usually like a water column where you're scooping up and aggregating across the water column, or you're turning your net sideways, dragging it, and you're sort of aggregating a large spatial area into one data point usually. But as we just saw, water quality is not uniform. There can be gradual changes, such as gradients based on diffusion and other chemical properties, and there are often discrete changes based off of confluence, unit boundaries, or inputs. And so because of that, there's a potential benefit to analyze zooplankton continuously. So if you're going to measure and analyze zooplankton continuously, there are two pieces you're going to need. The first is access. So very similar to what was just described, we use some sort of mobile sampling platform. In this case, we've got a modified pontoon boat with a flow-through design that allows water to be sort of sucked up at the front, go through some processing and kind of output. So access, check. We can drive through, we can use this boat to sort of go through continuous areas and use it as our sampling. But once you have access, the next piece is to actually measure these things. And so to measure zooplankton, we use a scan line camera with a shadow graph. So this is pretty cool. It's basically an LED array and a camera sensor with the water flowing right between them. So the sampled water goes through and a bank of LEDs sort of shines out and records sort of the shadow on the other side. And one of the really neat things about this is it actually preserves size. So you can see it doesn't really matter because the LEDs are housed with directional lighting. The target can be in the middle, and in this case, their shadow gets cast on the camera array. It's the black circle here, but it could also be closer to the LED array or further away, and it's going to cast a very similar shape or similar size shadow. So it's got this really nice quality where it preserves their size. And I'll sort of get into why that's important a little bit later. So with this, we can capture these really neat sort of these silhouettes, and they look something like this. So this is what the process generates. It captures this big silhouette image with a whole bunch of stuff in there, including the things we're after. So in this case, we're focusing on copepods, Daphnia or your water fleas, as well as amphipods. But because we're in a real world situation going through a dynamic delta, we pick up a lot of other stuff. So that can include a whole bunch of bubbles, a good amount of detritus and plant material, and every now and then you get stray small fish and other things like that. So we actually pick up quite a lot in these photos. And one of the things that makes this continuous is the fact that we get a lot of these. And as you just saw, having too much data can actually be kind of its own conundrum. So this thing takes pictures quickly. We're talking somewhere around 17 frames per second, which generates for a decent sized transect thousands of these images. And you can go through these by hand, but it is tedious and pretty unbearable. It's kind of like staring at TV static trying to find this stuff. So this all but necessitates a automated approach. So the other thing to keep in mind for an automated approach, though, is again, we're going through these dynamic environments, which means the conditions change pretty rapidly depending on time of day, water quality, and multiple other factors. So one of the first things we learned is that pre-processing an image is crucial, being able to sort of balance things out. And this is also going to help for our training data for the machine learning model. Speaking of machine learning classification, that's one of the main tools that we're going to be using here. And there's a couple of things to think about when we're going through this process. The first, our targets are really small, oftentimes maybe a dozen, maybe 24 pixels wide. So they're really, really tiny things in these images. And in general, doing a full image search is going to be very computationally costly on something this small. So trying to ask, where these things are in the frame is a pretty big task. So we found that it's actually a better plan to isolate the targets and then sort them that way. So this goes from where's Waldo to showing you a picture and saying, hey, is this Waldo? Which, again, as you can imagine, a lot easier to do, maybe less fun, but it actually saves us a lot of time computationally. So here's what that looks like. We take a raw image. So that's basically our starting input. We then enhance the contrast, try to separate those blacks and whites, sort of pull that color histogram apart. And from there, we use a threshold to sort of take the darkest darks and call them our targets. We then use those to isolate what we're going to call our targets. And here they're sort of circled in purple, although the purple's not showing up too well. But the point is that we're finding these targets back on the original image. And then once we have these targets acquired, we can run them through our machine learning classification model to actually put them into their appropriate buckets of copepod, daphnia, amphipod, or other. And then once we have that processed, we can enumerate them. So in this case, we've got 11 copepod and 53 water fleas in this one particular image, this one image out of thousands. So how can this be used? Well, see the previous talk, but also in general, looking at gradients and seeing that sort of spatiotemporal variability as we're moving into a different direction. So an example might be an upstream analysis, seeing how the abundance might change as you're moving away from a salinity or from the tidal influence and up into more freshwater areas. You can then aggregate these and compare across things. So now you can look at the variability in one transect, maybe you've got another transect, you can compare the variability between them. So think of maybe open water versus near shore. Obviously, this data serves as great fodder for other models. So if you have a habitat suitability model or an energetics model, they would love to have some spatial idea of biomass for energy. So again, it could be used as a nice input to other models. But in general, there's a really cool possibility where if you're measuring other things continuously, like chlorophyll or temperature or fish abundance, you now have sort of a continuous snapshot or representation of some minor food web interactions. So I'm going to show you what that looks like real quick with a couple of results. So as a very basic example, you can look at the abundance or the catch per unit effort of zooplankton count as you're moving upstream. These 4 different lines are color coded based off of pre-designated sort of unit boundaries. And you can see that there is some actual change or response difference in the zooplankton abundance across these different units. And that's just a quick example of that sort of upstream variation that can exist. And you can imagine if you're doing individual spots, you might be missing some of the high spots, or you might be aggregating things in a way that creates sort of that no man's land of a mean that doesn't actually exist anywhere within the actual environment. So if you do this multiple times, you might even be able to start building up that sort of location comparison. So here's an example of a heat map of multiple transects from one day showing zooplankton abundance. And it's kind of nice because it sort of comports to a lot of the hypotheses that we'd imagine. So locations that have more impounded, slower moving water tend to have a little bit higher abundance of zooplankton. Especially you can also look at sort of the difference between a near shore and open water, where there's more sort of availability for nutrients and other phytoplankton at the near shore, and you can definitely sort of see those patterns exist. And again, all of these are made of thousands and thousands of those images being sorted, classified, and sort of added up. And then finally, as an example of a food web, this is sort of like the gold standard that we imagine working with in the future, which is to take all of these all these different parameters on a continuous basis and being able to sort of ask questions of it. So in this case, we've got temperature, we've got dissolved oxygen, turbidity, chlorophyll, a relative measure of zooplankton abundance, as well as fish observations. So now we can start asking questions about where are we seeing zooplankton and fish? Where are we seeing zooplankton but not fish, and why some of that could be? And of course, this data set can then be sliced, diced, and turned into a whole bunch of very specific hypotheses once we sort of isolate the things of interest. So that being said, there are some pretty big limitations to the process. The first we've heard about a couple times today, turbidity. This is an imaging process. So when you have enough turbidity, it really wrecks the contrast. Suddenly the dark targets aren't dark and the white background's not white anymore. And it becomes very difficult for our models to be able to detect what's here. The second is debris. Even though the process of the scanline camera is really nice because of that feature where it keeps the relative size in check, there is still a 3-dimensional volume of water that we're looking at, and occlusion is real. So if we have a piece of debris flowing in front of something, that skews our counts. And so if you're looking at relative abundances and some areas have a lot more debris than others, you have to sort of make some corrections or think through that, or at least consider that as a caveat. Third is size. So zooplankton range in size quite heavily. We focused on some of the larger taxa, but then there are others that are much smaller. So something like a rotifer right now is far too small to detect using this setup. As you can see, some of the copepods were, like I said, a dozen pixels. That put our rotifers down to 2 or 3 pixels, and nobody's going to detect anything with that. And lastly, this is a pre-trained classifier. So we focused on the 3 taxa that I've shown you results for, but we can't detect something else until we know that we're looking for it. So something like a mysid shrimp or something along those lines, they're not going to be able to be classified in the current regime until we get enough training data and enough targets found within the camera to be able to start building up that classification. That being said, most of our future refinements seek to address these issues. So the big 3 are adding additional taxa. The more camera footage we get, the more examples we have of some of these additional taxa, the more we can build up our training data set and eventually classify them as well. The second one is a more compressed field of view. So if we were able to get that camera and that LED array even closer to each other, that should reduce turbidity because that gets worse as we have depth adding to it. And it should also limit the amount of occlusion that we have. So getting things squished together a bit more is going to be really, really useful. And finally, biomass estimates. Because we have that size preserving feature of the scanline camera, and we have a count of each of these things, there's only a couple hop steps and jumps to start turning this into a biomass. And if you guys went to the poster session, there was actually a really nice poster from SFU talking about the sort of carbon count in each of these particular taxa. So this is actually a little closer maybe than I was thinking prior to the poster session, but now it's looking like there might be an avenue. So those are sort of the big future refinements that we see as being moving this process along further. So with that, I would like to thank the folks that we worked with on this, including the Department of Water Resources, the Bureau of Reclamation, and the California Water Boards. And if you have any questions, I'm happy to answer them. Otherwise, I can take them later on my email.
And just please wait for the mic, please. Yeah.
Hi, what's your QC process? Do you have somebody like a ID expert to like go through these images, or are you just trusting the ID of the camera?
No, yeah, we do have a human-based QC process. We've got several hundred images of various resolutions and various quantities being measured by multiple humans so that we can get that sort of inter-rater reliability and compare them to the classification routine. We actually were quite pleased with the results where we're generally speaking in the 98 to 100% category. So it does a very good job of identifying the obvious ones, and the places it has errors are the places where the humans often have errors as well. So those sort of edge cases, it's still a little tricky on those.
Very cool images. You have such a push net set up. Is everything being filtered past this camera?
Essentially, the water that's sampled from the front of the sampling platform is all being funneled through and imaged through here. So yeah, I wouldn't say that it's, you have to be careful and not extrapolate too far because it is a small amount of water compared to obviously the channels that we're in, but everything does go through. So there's a chance for everything to be picked up.
Mm-hmm. So do you have like backflow issues since you're narrowing down from a larger wide space to such a narrow camera view?
Yes, but there's some, engineering that went along with this to make sure that that was minimized. And we also have additional sensors that allow us to understand when, for instance, the boat needed to back up or hit a different velocity speed so we can sort of normalize things and get a true catch per unit effort.
All right. And last question, if you don't mind. You're taking 17 images per second. How are you avoiding double counting organisms?
So that the speed has actually been set at such the reason we ended up with 17 frames per second is that it should allow essentially enough time for the camera to take the image and enough sort of the field of view shifts within that amount of time. If we're at the current or the correct speeds that we should be sampling at, then the math works out that each of the images essentially is a new set of water. If there was a truly mobile creature, though, a small fish or something like that, there's a chance it could be double counted. But generally, the zooplankton are a little too overpowered in these conditions.
Thank you.
Mm-hmm. Oh, sorry.
No, sorry. Thank you, Kai. Our next speaker is from USGS. Susan Dela Cruz will be presenting on the namesake of our session. I never told you.
Okay, well, thanks, Dan. And good afternoon, everyone. I'm really happy to be here today to talk about our Prop 1 funded project to evaluate the influence of tidal marsh restoration on prey resources, fish diets, and food web connectivity in the upper San Francisco estuary. We've recently completed processing all the samples that were collected for this study. There were a lot. So now we're busy analyzing the data and working on manuscripts, and I'll just be presenting some selected preliminary results. Definitely want to acknowledge all the co-authors on this study from USGS, from UC Berkeley, ICF, and state water contractors. It was a big collaborative effort and a lot of fun, and it's still going on. So region-wide restoration is underway in the Bay Delta to mitigate wetland loss and alleviate declines in primary productivity and native fish populations. And the guiding premise has been that this is going to increase food availability for native fishes. But we do lack some information on interactions among pelagic, terrestrial, and benthic food webs in restorations that it's kind of a central data gap to having us understand the effectiveness of this for species recovery. For example, previous studies have shown that restored wetlands can be as much as 50% less functional than reference systems, even many years post-breach, like 20 to 30 years post-breach. On the other hand, oh, I'm sorry. I really went ahead. Nope. On the other hand, these sites can also recover their biodiversity really quickly. So this kind of disconnect suggests that, structural food web recovery might not necessarily lead to functional recovery if you don't have some of these food web connections established. So because of this, understanding restoration effects on food webs has become a priority need for the IEP action agenda. So our study is designed to evaluate restored marshes of different ages in tandem with reference marshes to understand the effects of habitat evolution on food web development for fish from different foraging guilds. And we had 3 objectives in this study. The first was to measure prey abundance, community composition, and energy density at reference and restored sites. The second was to evaluate diets of fish from different foraging guilds at these sites. And then the third was to compare food web structure and energy flow amongst these sites. And that third objective is going to be discussed by Megan Pagliaro, who is doing this part of the study for her PhD. So I'm going to stick with the first 2 here. So this conceptual model highlights our hypotheses, showing that we expect terrestrial, benthic, and pelagic resources to vary with age and tidal influence in a marsh. And older marshes in the upper right are expected to have more total prey energy available for fish, and particularly from terrestrial resources, while newly restored marshes are expected to have mainly benthic and pelagic sources and less energy overall. So this is a really beautiful map by Amber Manfrey, and it highlights our study sites in the upper San Francisco estuary. We have paired reference and restored marshes in blocks along the salinity gradient. And this block design kind of helps control for large scale variation in salinity and environmental variables. And just quickly pointing out our restored sites, Tule Red is the youngest. It was breached in 2019, just prior to the start of the study. Wheeler was breached in the mid-2000s, And then Sherman Island was accidentally breached like in the late 1800s. So very, very old restoration site. So our sampling took place in the winter and spring of 2020 and 2021. We chose 6 random locations in the interior channels of each marsh and on the exterior edges of each marsh. And at Tule Red, we also sampled the interior ponds that were included as built-in features to the restoration to try to increase residence time and prey productivity. I want to just briefly go through everything that was collected at each sampling location. There were a lot of biophysical parameters that we'll be linking to try to understand drivers of prey productivity. Primary producers included everything you see here, including EAV, SAV, and FAV, which are emergent, floating, and submerged aquatic vegetation. Invertebrates were collected from multiple substrates, including terrestrial, benthic, epifaunal, and pelagic. They were ID'd to lowest taxon, measured, and then energy density was calculated for a taxon, which we had enough biomass. For fish, we used otter trawls and minnow traps to collect fish from multiple guilds and looked at diet via gut content and stable isotopes of nitrogen, carbon, and sulfur. So now on to results from our first objective here. So to compare overall prey production among sites, mean abundances of invertebrates from each habitat substrate were centered at 0 and then scaled. So these charts represent above average values shown here as positive bars and below average values. So in winter, you can see that Tule Red and Wheeler, the restoration sites, were kind of the ones that dominated prey production. Both had above average abundances of prey from benthic and emergent vegetation. Tule also had the greatest prey abundance from unvegetated sweeps, while Wheeler had the greatest prey from submerged vegetation. But then in the spring, we saw a lot greater productivity across the sites in general. Tule Red had similar patterns to winter, but relative abundances were greater. Wheeler patterns changed, that's the benthic and terrestrial prey abundance were positive. And Ryer, Chips, and Sherman had greatest abundances of epifauna on floating and submerged vegetation. And interestingly, Browns was kind of the underperformer with below average abundances for pretty much all prey types in spring and in winter. So now let's take a look at prey densities and community composition. We identified 118 unique taxa across all invertebrate samples. And to facilitate analysis and discussion, we consolidated the top 90% into 16 broad groups. And you're going to see these kind of reiterated throughout the talk. So a few things to note: that crustacean prey dominated the prey from all substrates. Crustaceans shown here in the kind of orange and red tones, except for terrestrial insects, which were springtails or columbella and dipterans. Let's see, Tule Red restoration produced high benthic and pelagic prey, but had less prey from terrestrial and aquatic vegetation, while our intermediate age restoration Wheeler, because the third bar from the left there, had the highest prey densities for 4 of the 6 substrates that we evaluated. So prey densities and community composition varied by season and habitat feature, interior or exterior, depending on the substrate that was sampled. So these examples kind of highlight this idea of the ecological portfolio effect, where you've got multiple habitat types that are networked networked environmental substrates that kind of maximize foraging for fish that are using the estuary through space and time. So you can see here invertebrates from submerged aquatic vegetation had the highest densities in winter in the interior versus floating had higher densities in the exterior in spring. So really a wide variety of availability. So we used multivariate methods to compare differences in prey communities. Primanova indicated prey communities varied by season and habitat type, kind of like I just showed you in the previous slide, but not by restoration status. So overall, not by restored versus reference. And we used a non-metric multidimensional scaling to examine more closely the similarity and differences among all 6 study sites. Okay, I got to speed it up. Paired restoration and reference sites are shown in different shades of the same color. And so one thing to note here is that 2 of the paired axes plots, truly red is sitting out there all by itself. And so it doesn't share any overlapping polygons with the rest of the sites, indicating that communities are very dissimilar, even between its paired reference site. Which is, we think, kind of indicative of the lower prey productivity in communities that we saw at that site. So results for fish diets, speeding up. We evaluated 890 fish from 16 taxa that were the most numerous and found at all sites. And of the fish stomachs dissected, 90% contained detritus, 57% contained invertebrate prey from 56 unique invertebrate taxa. And you can see they're categorized as the prey was. Crustaceans dominated the diets, especially for pelagic and demersal fish. So to evaluate our hypotheses on diet composition, we combined diets of fish within their associated foraging guilds. Overall, pelagic fish had higher numbers of individual prey items in their stomachs. Mean prey abundances in fish across guilds were slightly greater in reference compared to restored wetlands, while mean prey abundances in pelagic and benthopelagic fish collected in exterior habitats were just slightly greater than those for interior. And the reverse was true for demersal. Another thing to point out here is that all guilds had greater numbers of dipterans or flies, which are terrestrial prey known for their high energy density in reference in interior habitats. So to visualize relative importance of different prey taxa, we looked at prey occurrence and abundance in fish from different foraging guilds at restored and reference sites by plotting the percent frequency on the x-axis and the percent prey specific number on the y-axis. So taxa that are the most important are in the upper white quadrant of this plot because they're found in high abundances and in large proportions of the fish. Okay. So you see that nothing is in the right quadrant for fish from restored sites, but we did have some standout taxa that were approaching the right quadrant. So copepods for pelagic fish, nematodes were most important for benthopelagic fish, and amphipods for demersal fish. But at reference sites, we really saw these taxa standing out in the right quadrant, at least for benthopelagic and demersal fish. Again, we used multivariate analyses. Primanova indicated that prey consumed varied between seasons, but not by habitat or restoration status. But then when we looked at these paired plots, we're showing no overlap between the diets of fish overall in reference and restored sites, Wheeler and Chips pairs and Sherman and Brown's pairs. And interestingly, Thule Red and Ryer diets did overlap across all seasons. So in summary, while prey densities were highly variable across study sites, we found no difference in community composition when looking at just reference and restoration sites overall. But restoration age may play a role in community development. Truly red prey communities were dissimilar from all other sites and had relatively low densities of invertebrates from terrestrial and submerged aquatic substrates, perhaps reflecting their limited vegetation present at this early restoration site. Truly red did have greater prey densities of from benthic and unvegetated substrates, even than older restorations, kind of supporting the idea of a trophic surge in which early restoration sites can be very productive due to nutrient inputs from adjacent habitats. Oh, and my time is up. Okay, one more thing about fish. So mean prey abundances and diets of fish from all foraging guilds were greater in reference marshes, suggesting that prey may not be as available or as accessible to fish in restored marshes at this point. And I think I'll leave it at that because I know we're going to go, but I did have a slide about next steps and just want to point out that we're still in heavy data analysis and a lot more that will come out of this study. And thank you very much. I'll turn it over to Megan.
Okay, so next up we have Megan Pagliaro, and let me just get yours up. Okay, you're off.
Hello, everybody. As I said, my name is Megan Pagliaro. I am a fifth-year PhD candidate at UC Berkeley in the Ruhi Lab. I'm very excited to talk to you today about my PhD research, which is in collaboration with USGS, as Susan just so amazingly described, as well as ICF. And this project is funded by CDFW's Delta Prop 1 and the Delta Science Fellowship. So to get us started, we still don't really know whether global efforts to restore wetland ecosystems is truly working. So for instance, in this meta-analysis by David Moreno-Mateos and others, it shows that biological structure and biogeochemical processes can be pretty hard to recover. And in some cases, it can take 50 to 100 years, as Susan was alluding to earlier. So I hypothesize that this may be happening because even though we see like key biotic components returning, the actual energy flows and connection within that food web is not being restored. So I centered the main question of my research to address this knowledge gap. So specifically, I'm asking, does the restoration of tidal marshes translate to the recovery of energy flows and food web structure to similar levels of those of reference wetlands? And I am addressing this question in the Sassoon Bay. So it's a map so pretty, you get to see it twice. So as Susan described in detail, so I'm just going to do a brief overview. We studied 6 sites, 3 reference in orange and 3 restored in yellow. We define a restored marsh as one whose dike or levee has been breached, while reference marsh is one that's never been cut off from its tidal flow. Analysis is based on a block design, each block consisting of 1 reference and 1 restored tidal marsh. We've already completed 4 sampling seasons in winter and summer of 2020 and 2021. And so in order to measure these energy flows and food web structures, we collected samples spanning from the basal resources to top predators and processed them for stable isotope analysis. So we collected carbon, nitrogen, and sulfur data on all components of the food web, but for this talk, I'm just gonna discuss on results from our 808 fish and 80 FPOM and phytoplankton samples. And I'm gonna go through what each of these isotopes measure for us. So carbon helps us parse out the different contributions of different basal resources. So for example, diatoms and phytoplankton often have a more negative delta 13C seen in that top figure in green. While the brown or detrital pathway, mainly indicated by decaying marsh plants, have less negative values. For nitrogen, nitrogen tracks the trophic position of the topmost predator, and well, it moves in a measurable way as you move up the food web. And this is about a 3.4 change per mil as you move up each trophic level. With that said, that value gets very finicky when you have a lot of omnivory in your system, like ours. Finally, for sulfur, sulfur present in the oceanic water column, and that's highlighted in pink, is much more highly enriched. So it has plus 20% parts per million, while sulfur found in freshwater sources have a much lower enrichment value of plus 0 to plus 15 parts per million, and that's in green. So we have these three isotopes to be able to compare and track energy flows in the system. And I'm going to use this in two main ways. So with the nitrogen isotopic data, I am going to calculate food chain length, or the number of links between the basal resource and the top predator. And with all three isotopes, I can calculate the isotopic niche volume. That is the volume that encompasses all three of those values for a community of consumers. And this is graphically illustrated in that lower figure by Rossman et al. And this was done using the R-Package niche rover to calculate the hypervolume. So moving on to putting these metrics into practice and getting some actual results. So starting with my first goal of examining food chain length in tidal marsh pairs, we hypothesized that food chain length in reference marshes will be much higher than those in restored. And so here's a figure of those results. We calculated food chain length by first calculating the maximum trophic position for each site in each sampling season, and then using FPOM and phytoplankton as that base to subtract from. And the fish with the highest average delta 15N that's what species was considered that top predator. So what you're looking at is at the bottom is the type of site. So reference in orange as with the map, and restored in yellow, again, as with the map. And then we have each pair. So each of those three blocks, we have pair 1, pair 2, and pair 3. And then that was looked at for both across all sampling seasons and then split up in 2020. 21. So the overall trend is that food chain length was higher in reference sites compared to restored, as we hypothesized. When looking within years, there is a little variation with P1 not being significant in 2020 and P2 not being significant in 2021. So it does show that there is like some variation within years, but and so we wanted to, again, look further into how this isotopic data can help us know more about these communities. And so next is the niche volume. So I'm looking at the variation in the isotopic niche volumes first across all of our marsh sites. And then I'm specifically going to be looking at comparing native and non-native species. So for the whole community, we hypothesize that there's going to be smaller average isotopic niche volumes in restored versus reference tidal marsh sites. And so we calculated the isotopic niche volume for each fish community at each site. So again, at the bottom is the type, reference and restored, orange being reference, yellow being restored across each pair, and then separated into the two different years. And on the y-axis is that niche volume calculation. So the numbers dictate the results of Cohen's D effect size analysis, and they range from a small effect size in light blue to a large effect size in dark blue. And the larger the effect size and darker the color is, the more significant those differences are. So restored sites seem to consistently have higher niche volumes than, restored sites have lower niche volumes than their reference. You know, you say it enough, it gets mixed up in your head, but with one exception, and that is in pair 3, both across both sites and in 2020. Pair 3 involves our youngest restored site, Tule Red, which was breached in October of 2019. Then sampling started beginning in January of 2020. So if you see in that first year in 2020, the restored site has a higher niche volume than its reference. But then in that second year, 2021, that flips. And the references now has a significantly higher trophic volume than it's restored. So we think that this is evidence of the trophic surge hypothesis, as Susan was discussing earlier, that influx of decaying plant material and like flooded vegetation has this surge of nutrition that artificially bumps up the community composition until like succession dynamics and competition take over. So additionally, we wanted to know what was driving these differences, whether it was species diversity or species abundance. And so we ran a resampling analysis on the data to look at what could possibly be driving those differences. So C and D is the resampling analysis controlling for abundance, and E and F, those lower two, are the resampling analysis controlling for diversity. And as before, niche volume is on the y-axis and whether the type is on the x. So when controlling for abundance, or there's the same number of individuals in each pair per year, the trend remains. We see very similar trends as that first niche volume calculation. But then when we control for diversity, so there's the same species per pair, the differences are a lot more muted, as well as the significance of those differences are very low. So from that, we hypothesize that it's species diversity that's driving these differences and these patterns. So moving on to the final component of my talk, and that is looking at how the niche volumes of native and non-native species compared. So I wanted to look at generally comparing these differences. And so this is across the whole system as a whole, not within pairs. And so I did this by looking at both the niche volume as well as the amount of overlap of that volume between the native and non-native or introduced groups. So figures A and B show the differences in the overall size of each group's niche volume across all sampling years and then separated in sampling years. And if you see at the bottom, so you have gray is the introduced species, red's the native species, niche volume is on the Y. And then for C and D, that is looking at that overlap percent probability. So on the Y is that overlap probability. On the X, how the best way I can explain this is by just going through. So in that first gray in C, that first gray plot, it is looking at the percent probability that an introduced species is found within the niche of a native species. And then that the red is looking at the percent probability that a native species is found within the niche of a introduced species. And then again, those are separated for 2020, 2021. So what does this data mean, like visually? That introduced species have a higher niche volume, but a much lower percent overlap probability. And here's a little figure I came up with to show what that means. So there is high overlap in the two groups, but there is asymmetry in that overlap. And it is clear that introduced species are occupying niches that cannot be occupied by native species. But whether that is because they outcompeted and took over a spot or took an empty spot is not known from my specific analysis, but is a interesting thought to continue forward. And so in final conclusion, so looking at the niche volumes, the restored site Thule red actually performed better than its reference site in 2020. It is species diversity that is driving these differences in niche volumes. And some non-native and native species may be occupying separate resource pools. And so for our next steps, we have a bunch of isotope data, as Susan was saying, and it's really exciting to continue to analyze it. Next, we're going to look at the mechanisms that are actually driving these differences, as well as start doing some really cool synthesis across the project with all of our results. And so with that, I'd like to again thank my funding agencies, the Delta C Grant Delta Science Fellowship, as well as the California Fish and Wildlife Delta Prop 1, my amazing collaborators at USGS and ICF, and of course, there's many other special thanks. And I'll take any questions.
Hi, very interesting talk.
Thank you.
There seemed to be a lot of variation between years and between sites, you know, even within sort of a category, like the same site would have niche volume of 900 one year and 600 the next year. What do you think was driving those big swings?
Salinity. Because I think 2020 was an unusually dry year while 2021 was unusually wet. And it was interesting. We were actually able to see, so at Tule Red, which is like the most salty site, versus Sherman, which is the closest to confluence, the like invertebrate pools that we'd find at Tule Red year one, no, it was Sherman year one, we're trying to like Tule Red year two. Anyway, it like flipped. So I think that salinity is a big driver of these communities as, and so like the differences in the water years clearly did have a big effect. Any other questions? Hopefully I answered what is an isotope a little bit. Please.
Thank you. When you were showing the mean food chain length of the different fish species, were you able to parse out whether that's due to composition of the different fish species that causes that difference in food chain length between restored and reference site?
So it is a specific species that was used to calculate that. So what, how we did it is I essentially just lined up all of the species by their delta 15N values. So that's that trophic position value. So the higher it is, the higher they are in the food chain. So I got the average value of that for each of those species. And so whatever species had the highest average was considered the top predator. And so that is just the like different, all those different species, their different like food chain lengths is what makes that plot. Does that make sense? But yes, that is the next, like which species do that is very interesting, and manuscript coming soon if you want to know more about that. Okay. Thank you very much.
Thank you, everybody, for attending the session. The next session begins at 3:30, so make sure you're back for that.
Paul, you hear a lot about cyanobacteria and about the toxic blooms and how dangerous they are and what they are doing. But cyanobacteria are actually very amazing microorganisms. I want to open it with that. They have very unique physiology. They are the first organisms that were able to fix the nitrogen. So that's why we have the oxygen atmosphere like we have today. Many of them, they can fix the nitrogen as well. And they can come in many different shapes, from single cells to colonies. They can be multicellulars, and sometimes you can even see them by bare eyes. Another advantage that they have is that they really can control the movement in the water column. They can also do sliding, and they are like kind of always finding their way towards the base conditions for them to live. And basically they can be found everywhere. As you can see here, I posted some like example pictures that they are found in cold system, in hot system, even in the deserts. And if you've seen something like that on your hikes, this is all cyanobacteria. But also they are often like forming these huge aggregates in the lakes and the river systems that they can be seen even from the satellite, by the satellite. But these cyanobacteria are also very interesting from point of view that they can have a different secondary metabolites. Many of them, maybe you heard about spirulina, that is kind of like food supplement, or they also have some specific metabolites that can be used as a sunscreen. But once again, they can produce like second part of the compounds, which are often toxic, harmful to animals, harmful to humans. And they are disturbing ecosystem balance in general. And we know like what and when and how these cyanobacteria appear. And it's starting to be more and more frequent all around the world because we are having increase of the temperature, which leads to stratification, like somebody was talking today. And basically we have the longer periods that these cyanobacteria can proliferate. Generally, planktonic cyanobacteria are much more studied, but there were like some recent cases. I don't know if you've seen it in science about the eagle killers. These were like the cyanobacteria producing compounds, which were literally killing the eagles through the cascade. And it was not planktonic, it was growing on invasive species of some plants. But most of all, cyanobacteria, they don't live alone. They always attract specific bacteria around them. And it's kind of also important to look at them as an ecosystem rather as a single organisms. So in the delta, this cyanobacterial bloom has been studied for the past 20 years. So we know, basically under the leadership, Peggy Lehman, they did a lot of studies. As you can see again here, like there was many sites that they've been, monitored regularly. They were testing many different things that they really pioneered the research of cyanobacteria and the cyanotoxins. And they found many environmental conditions that are actually benefiting the bloom of cyanobacteria. Mainly the focus in the past years at the beginning of the studies were on the one cyanobacteria called microcystis and the one specific toxins that they were producing, which we call microcystins. And these microcystins are basically found all over the world, and there have been like some effects that they had on the human health. And so that EPA came with the, the levels of the toxicity of these toxins that should not be passed if we want to consider the water safe for drinking. And as you can see here in this map and in this slide, the area around Stockton in 2020 reached really danger level of these cyanotoxins. But however, by the time there was not only one toxin present in the delta, there started to be like other toxins present, and also not only microcystin was present there. There were like other cyanobacteria which are having potential to produce the other toxins also appearing in the delta system. And here I just want to show you like how they look like. These are the samples that they collected the last year, 2023. So they can be these unicellular microcysts that you might have heard of, that they are forming really these dense colonies, or they can be the filamentous forms, and even having these specialized cells that they are helping them to fix the nitrogen in the low nitrogen conditions available in the water. So they basically just take the nitrogen from the atmosphere and get their own nitrogen sources. And the third one is another filamentous cyanobacteria, which is also appearing in the delta system, already being reported in the work, again, like under the leadership of Peggy Lehman. Not only that they were like appeared from 2014 to 2017, the great work of USGS, like mapping the more of the South Delta, also recorded like mixed cyanobacterial communities across the different sites. And they measured the different cyanotoxins. At this specific time, I think that was like 2021 year, they detected 3 different cyanotoxins with a different range of concentration. So basically, many things is known in the delta. Like what is the novel is like, we have new cyanobacteria, we have new cyanotoxins, but I was like, what are these cyanobacteria doing there? Is like, are they having actually some genes there? Can they produce specific compounds? And if they can produce something, what they're actually producing it? And like, what is going on with all these bacteria surrounding them? Like, are there some like specific bacteria always growing within these cyanobacteria? And are they helping them also to kind of like overcome some environmental conditions which are maybe not that best for the cyanobacteria to grow? So for that purpose, getting to know like the delta system, hearing about what is going on, and attending last 2 IEP conference, like virtually, I got to know that there is like some lack of knowledge that maybe we could address it by basically linking the microbial diversity around these novel cyanobacteria and actually see like what the cyanobacteria are producing. So there were like 3 major objectives. One was actually to see the composition of the cyanobacteria bloom. and see the toxins that they can produce. And for that, we would use like technique where we would extract the DNA of all the organisms which are there and reconstruct metabolism of each of these microorganisms that we can find. And this basically will allow us to say like, there is this cyanobacteria and we know the full gene set that they have it. And in this gene set, they have the genes for specific cyanotoxins to be produced. And we combine that also for the actual measurements of the cyanotoxin throughout the delta. And at the end, like, kind of like, what is going on with environmental conditions? What is driving like this completely interesting, diverse cyanobacterial community and the cyanotoxin potential? So when we wrote the project proposal for the fellowship, I had a huge support from the beginning. First of all, it was Jill from the UC Berkeley, Keith as my community mentor here at USGS, Ted Flynn from EMP crew, like basically they really let me go on the boat and sample along their monitoring effort. Judy, who did all the cyanotoxins, Sylvia, who was like, I was talking always constantly about how to approach the cyanobacteria sampling analysis. Xavier from National Lab and Healy that I met along the way, basically, who were doing some other studies and it was kind of complementary. So basically what we done, there was like 3 kind of different sampling efforts, and I will be presenting one of them. So first one was regular monitoring at EMP. So I collected 80 samples of that, and then I joined the USGS crew in July and October, where I collected 90 samples, and then I got also samples from Discovery Bay and Stockton. Before going to perform actually all this sampling, I did the pilot sampling to see if my strategy would work. There is like amazing USGS crew who kind of like allow me to join the boat and just collect the samples. And the regular sampling over 8 months was on the Sentinel, where was really kind of like thought through, and this is the results that I'm going to be presenting you. And of course, like huge metadata from both groups. So I will kind of like rush through what I have done. So we did the cyanotoxin analysis, 16S, meta-barcoding, full genomic, metagenomic sequencing. And then we also did some nanosims analysis, non-targeted LCMS, and also like some another sequencing technique. What we found in 2023, which was unusually wet year, so there was not really the cyanobacteria bloom, which was interesting to kind of like approach it from the different ways. Like, I want to study cyanobacteria bloom and there is no bloom. But luckily we kind of like really concentrated the biomass and we got interesting data. Generally, the trends were kind of similar throughout the sites. I would just point out the south site, which is MD10A and P8, which turn out to be quite unusual ones. When it comes to cyanotoxins, out of 22 that we tested, we detected 16 of cyanotoxins. And the most common one were microcystis, but some of them like anavenopeptine are also present across the samples. When we look at the concentration, it's like, you know, when you're making the plots, everything, I didn't want to mess with the scale. So I just want to show you how it looked like over the samples. But on the other hand, I also wanted to show you the relative composition of these toxins. What is the most striking that in the early months as April, we already found the toxins. Like our anatoxins are the one that people are not usually talking about. And then throughout the season, we have like different variants of the microcystis. And then in the P8, like Stockton site, we even have like some very interesting mix at the end of the season in November. We looked at phytoplankton composition on microscopic data. Cyanobacteria didn't dominate. So this is the data from EMP, so from the one non-concentrated cyanobacteria samples. But luckily we got like quite good representation of the cyanobacteria community. And we look at the microbiome. So we can see the seasonal changes where cyanobacteria starts to dominate in July. It goes through November. This was supposed to be very nice UMAP, which would be showing you how we had a nice grouping of our microbiome starting from the April circling down to November, which is showing us that the microbiome that we collected is actually really like related with the month of the sampling, which was also the result of the permanent testing that I did before. And finally, what about the cyanobacteria? So at the moment, I'm showing 16S results, the metabarcoding, because genome-resolved metagenomics, it's taking a lot of processing time, so it's being processed. I hope in 2 months I will have the data back. And what we can see, like shortly, is that we have the co-dominance of the microcystis and the afanisomenum. So the blue ones are afanisomenum, these are the filamentous nitrogen fixing ones, and the red one, it's microcystis, but we have appearance of some another unusual cyanobacteria. And I would just point out like what struck my kind of mind or like what pay attention, because these data are really freshly out of the oven, is like these two sites. So these two sites showed kind of like interesting diversity. First of all, at this site, we had the highest concentration of the cyanotoxins that we found. And if you look at the composition of the cyanobacteria, it's very diverse. It's basically composed of like 3 different cyanobacteria that we know that they can produce cyanotoxins. And secondly, this interesting peak in October and November, it seems that belongs to Tychonema, which is benthic cyanobacteria. And we find in these cases like 3 different toxins. And most likely the producer of the toxin in this case are actually benthic ones. So I would conclude that I just gave you really like small snippets, but just to kind of show you like what we found in this even unusual, not unusual, but like wet year, like using our technique. So water condition definitely affect the microbial dynamics and the cyanotoxin diversity. And my time is up. Thank you. I need to do last. I have to thank EMP and Sylvia. That was like amazing having me there for 8 months. Every month, everything was just perfect logistically, and like it was very welcoming crew. Thank you all.
Ed, is your presentation on here? It's not in the right order.
Oh, there.
Oh,
Yeah.
okay. Sorry. Do you want the pointer? Sure. Okay. So our next talk is Ed Gross, and he is with Research Management Associates. They recently been incorporated into another company, so I wasn't sure. Ed.
Okay, thank you, Ellen. Okay, so I'll be talking today about the contents of this paper using age tracers to estimate ecological rates in a phytoplankton model. And I'm a hydrodynamic modeler, as are several of the other people on this paper. And it's really more of a method of introducing a new modeling method than it is, reporting specific results. Although we do have some results to report. And this was done on Wim Kimmer and John Duran's projects, 2 different projects spanned. Okay, so some of the ecological research goals are to quantify some mortality rates for in a phytoplankton model, clam grazing, zooplankton grazing, and then other mortality, a couple other mortality parameters. And to quantify spatial variability and growth and loss within our model domain of phytoplankton. Okay, so yeah, the main thing I really want to emphasize in the presentation is that this is a different approach, and it may start to sound like an approach but it's probably not. It's only been a few papers so far that have used this approach. Only one that I know of before we applied it. So you could think of it as related to two extreme sides of modeling. One is a box model where you have no spatial variability in your model. You just have a single box, or you could have multiple boxes, but in the simplest case, a single box. You'd even do sort of a back of the envelope calculation in your box model. So those completely ignore transport generally. And then you could have the really computationally intensive option of a coupled hydrodynamic and ecological model, where you compute them both on the same spatial grid and same time steps. So for example, our San Francisco estuary grid is about 1 million grid cells, and we calculate transport at a 60-second time step. So the best in some ways option would be to compute your ecology at the same time in spatial steps, but it's extremely computationally intensive. So what we're trying to do is keep as much as we can of both of those, the good aspects of both of those, keep as much of the speed as we can of a box model, but have a useful representation of transport still captured. So I want to emphasize this is not like a just offline, running a model offline, running hydrodynamics and saving a huge file and then running ecological model with that huge file. We're not doing that. We're not doing grid aggregation. This is totally different. It's the Lagrangian model along the lines of a Streeter-Phelps equation, which I'll get to again later. So it's a very simple model. This is not a simple model. This is in contrast SFEI's Delbach approach. So this is a great approach, but the limitations, potential limitations of this, we already discussed the computation, but also there are a lot of parameters that go into this model, and some of them could be site-specific, varying in space throughout the estuary or whatever study site you're looking at. And it can be very hard to understand, whether you've really chosen the right parameter values. So if you know all the relevant rates and other parameter values, then this, of course, is the right way to go. And what we're exploring in this presentation is really more useful if you don't know the rates and you want to explore those very quickly. And it certainly does not replace this sort of model. It could be used in conjunction with a coupled model to feed into it rates that you can very quickly explore parameter space, and then use what you've learned very quickly to inform this more computationally intensive model. Okay, so this is the approach we're applying. We run our model, our untrimmed model, and our domain, as we'll see, will be the Sassoon Marsh. And that model run over several months, takes about 25 days on a pretty powerful workstation computer, not a supercomputer, but a nice workstation. And we compute from that a bunch of tracer fields, which I'll define in the next slide. And we pull those out of the model only at the times and places where we have observations that we want to compute chlorophyll at. So we're taking out of these several terabytes of data generated by Untrim, we're pulling out a very small amount of information that you could store in a few kilobytes that you could put in a small spreadsheet, for example. And that's all we're using in the phytoplankton model, just that extracted information. And then we're feeding in some environmental information, some measured information to feed the growth formulation. So that includes solar radiance, temperature, and turbidity. And then we can use some starting parameter values or parameter ranges from literature to start off optimization. And then we run the phytoplankton model, which instead of taking 25 days, takes 0.3 seconds. And we can, because it's so fast, we can run that in an optimization or Bayesian inference framework. And we can quickly determine whatever free parameters we have left in our model, whatever we haven't specified, we can estimate the parameter values that allow us to best fit the data set that we're comparing against. Okay. So we're using several tracers to do this, and I'm going to define those really briefly. So here's a figure from Lisa Lucas and Eric Deluer-Schneider's paper. Really great paper to learn about this stuff. So the first thing is you could ask with, and you could answer with the tracer, is how much the water at any location came from a certain source. So in this case, you could tag the river water with a concentration of 1, and then you could look at the concentration anywhere downstream, like for example, at this point, X1 at time T1, and say, what's the concentration? If the concentration is 0.5, then half of that came from the river, and half of it came from somewhere else, like the ocean. So that's one thing you can do with tracers. Another thing you can do that's until the fourth thing I'm going to discuss, these have all been used extremely widely, these tracer metrics. So you can calculate the age. So the age you can think of as an average travel time from this X0 to X1. And, you know, since we have tides in the estuary, it varies in time also, not just in space. And then you can ask more detailed questions, like you can say, of that time that the water has been in the estuary, how much time did it spend in a marsh plain spatial compartment? And you can also ask the question, just more recently, as of 2019, an approach in a paper that I was first author of, we proposed this method of property tracking, property exposure. We can ask questions like, what depth did it experience when it was in marsh plain? So what average depth? So these are all averages. So we don't know, for example, you know, traveling from X0 to X1, two different molecules of water could follow two different paths, or, you know, and when you look at a bucket of water at X1, you have a range of water molecules or particles that have followed different paths and have different ages. But what we're calculating is the average of all those, the average age of all those molecules in the bucket. And these are all averages. So these are the equations. They're basically just an advection diffusion equation with source terms. So they're very simple to implement in a hydrodynamic model. And here's our model domain in the polygon. It's Sassoon Marsh, and we have boundary conditions at National Steel, just north of the Sassoon Marsh salinity control gates. And our downstream, or seaward boundary condition is in Grizzly Bay, where we have chlorophyll measurements and water level that we're going to specify. So then we have 4 stations shown here at Sheldrake, First Mallard, Hill, and Peytonia, where we have chlorophyll data. We also have turbidity and temperature that we're going to use. So we ran that hydrodynamic, it's a 3-dimensional hydrodynamic model. All the fields I'm showing are going to be depth averaged, but the model itself has 3-dimensional hydrodynamics. And these are the spatial compartments I mentioned. So we have 4 spatial compartments. We have a main channel and a side channel and low intertidal and marsh plain. So they're mostly defined based on bathymetry. And the reason we have 2 different channel compartments is because of a paper by Baumsteiger et al. They found more clams in the area we're defining here as the main channel. So we wanted to be able to allow the model to choose different clam grazing rates in those different spatial compartments. Okay, so here on the left is a snapshot in time from the tracer simulation, showing some of the fields, not all of the fields that are calculated. So we have the water fraction, which is the percent of water that came from our boundary, which is came from our downstream boundary in Grizzly Bay. So this is saying what percent of the water at any location came from Grizzly Bay during the simulation. So if it's less, wherever it's less than 1, it means either that some of the initial water in the domain is still there, or some of that water came in from another source, like it could have come in from the other side by National Steel. So then we have the age, the time it took to travel from Grizzly Bay to any location. And then I'm showing just for one of the compartments, the things that were one of the spatial compartments, one of the 2 things we're calculating. First is on the left is the exposure time. Can't actually see this. Let me make sure I'm reading the right thing. Yes, that's the intertidal exposure time. And then on the right is the depth that it experiences when it's in the intertidal habitat. And then on the right side here, we have showing it at each of those 4 stations that I mentioned in the previous slide. Well, 2 back. We show the full time variability of these properties at all 4 spatial compartments. Okay, and then we use a growth formulation from Klern in 2007. I'm not showing it here because it's a little long and I'm pressed for time. But we don't change anything there. We just use the parameters from Klern. And then the unknown parameters are shown in green under the losses. We're treating the phytoplankton mortality and clam grazing as unknowns, and this KP parameter in the bottom, which is sort of a nonlinear feedback parameter, Counting for things like you might get, non-linearly higher zooplankton grazing at higher phytoplankton concentration. So something like that could be roughly accounted for by this KP parameter. And then we do not have, this is not even an NPZ model. We don't have we're not tracking nutrients because we're assuming only light limited growth, and we're not tracking zooplankton, mostly because we're using an equation from Kimmerer and Thompson to give us the microzooplankton grazing rate. So that's shown here. And I'll mention actually just in passing here that we actually allowed the two constants in that equation to be variable and fit those, and came out to almost exactly these numbers. So that was encouraging. Okay, and then we're driving it with some observed data. So our boundary condition is chlorophyll in Grizzly Bay, observed at the USGS Grizzly Bay Station. And then we have some data from UC Davis on light attenuation and temperature is driving the growth formulation, and some incident light data from the NOAA NER. Okay, and then these are the results of our model. So on the left is the box and whiskers plot showing the range of chlorophyll at each station. So of course the boxes are the interquartile range. The line is the median. So we generally predict the right range at each station, except we're missing the upper end at Sheldrake. And as you can see on the right, for a short, this is a subset of the time period. We're simulating about 2 months in this model. With the 2 months when data was available. On the right, so it's more visible, we show just a couple weeks. And you can see that the tidal phasing is also predicted reasonably well by this approach. And we missed the high values at Sheldrake. We're not totally sure why, but perhaps because there's some managed ponds leaking high chlorophyll water into the slough that we're not accounting for. Okay, so then you can also look at how each of these terms affects the predictions at each station. And this is maybe the most important point of the paper, that the time, more time spent in the main channel is not a good thing. So, when you talk about residence time, it's also important to ask, where the phytoplankton is spending its time. Because in the main channel, there are net losses due to clam grazing. And in all the other three spatial compartments, there's net growth. So really, the important thing is to spend time outside of the main channel. And the rates that we got in our fitting are shown on the right. Side channel grazing was fit to be 0. Okay, and then this is what we consider a validation data set. It's not validation in the sense that it's a different period. It's actually the same period, but it's a very much larger spatial extent that we applied the same rates that we determined from fitting that time series data. So you can see the observed, this is from USGS underway data, observed on the left and then predicted on the right. And you can see on this bottom right panel, you can see that, somewhat of a good predictor of the chlorophyll is the time outside of the main channel, spent inside the domain, but outside of the main channel. So you generally have higher chlorophyll with time spent outside the main channel. Okay, so conclusions. We can predict the observed chlorophyll pattern during the study period. And the strength of this modeling approach, again, it does not replace the computationally intensive coupled model, but it does allow us to very quickly test model formulations, fit rates, and gain some conceptual understanding. It's much harder to understand the contributions of individual processes in the full model because there's so much more going on. Okay, and then I guess I'll just mention the bottom bullet. There are other manuscripts using similar approaches. Yeah, and thank you.
Okay, running out of time. We all have good talks. I'm going to be giving the third talk. So I'm Ellen Priest with Department of Water Resources. And this is a project that I did in collaboration with Janice Cook at the Central Valley Water Board and Tim Otten and Catherine Cook at Ben Genetics. This project was funded by the Delta Regional Monitoring Program SEP funds, the State Water Board, and then the Central Valley Water Board provided in-kind staff and boat time. And we looked at how benthic overwintering cyanobacteria, specifically Microcystis seed stock, the role that it plays in delta bloom dynamics. So first, I want to give an overview of the characteristics of Microcystis that we see in the delta. On the right is a conceptual model or schematic that we developed for the Delta CHAB monitoring strategy. And we categorize Microcystis habitats into 3 categories, roughly. The first is panel A on the lower right-hand side. These are our left-hand side. These are delta dead-end channels and marina areas. This corresponds with the picture in the upper left-hand corner, Stockton. So these are areas that the euphotic zone is throughout the entire water column. They're shallow, they're warm, they have long residence times, and Microcystis blooms really aggregate here. And you can see in that picture that you have this oily green scum that's characteristic of a lot of Microcystis blooms around the world. Panel B, this corresponds with the main channel environments. So these are deeper areas, up to 12 meters or so in depth. The euphotic zone is shallower. It's only penetrating through the top portion of the water column. We have more tidal velocity and river flow, and so the water column is more turbulent. The picture on the bottom here is the San Joaquin River, and this is an example of one of these main channel habitats. You can see Microcystis is still present here, but it doesn't have the ability to aggregate in the same way that it does these hotspot or dead-end channel locations. Finally, we have the flooded island sites. That's panel C here on this schematic. These are not as deep as the main channel sites, but they're deeper than those dead-end channels, marinas. There is more velocity and turbulent mixing than the dead-end channels, but less than those main channel environments. And then panel C in the upper right-hand corner is Mildred Island, and it shows an example of how Microcystis presents in one of those habitats. You can see that it's more aggregated than in the San Joaquin River, but less so than the Stockton. It's also noteworthy that there's other cyanobacteria species in this panel C. So this brings us to what do we know about Microcystis life history and what's the knowledge gap? We know that when water column is warm, 20 to 30 degrees Celsius, Microcystis can thrive in the surface of the water column. When the light starts to dissipate later in the year and our water temperature cools, those cells can settle into the sediment and overwinter until conditions are right for them to inoculate the water column again. We don't understand what the contribution of these overwintering cells or the seed stock is to blooms in the delta, and this is important for management. We don't know where blooms originate. Do Microcystis develop and grow throughout multiple areas within the delta? Or are they originating in locations with longer residence time, those dead-end channels, marinas, which we identify as hotspot locations, and then spread by advection, tidal movement through the delta? So we have 3 hypotheses for this project. First, that the static peripheral areas or hotspots, Discovery Bay and Stockton, will have the highest overwintering Microcystis seed stock. This is based in part on those pictures that I showed you, where you could see that thick aggregation of Microcystis in these areas. And then also this heat map on the right that shows chlorophyll at the Stockton waterfront. This is about a 5-mile transect. This is the terminus of the channel, is the public dock on here. And you can see the chlorophyll signal, and this entire chlorophyll signal is generated by Microcystis in this figure, is much warmer in the dead end area, and then it cools as it moves into the deeper environment. Our second hypothesis is that these sites are the primary sources of Microcystis in the central and southern delta, and that these sites will experience earlier recruitment to the water column and larger blooms. So we have 8 sites that we looked at Microcystis seed stock, and then we expanded that to 12 locations, which are shown on this map, to see how Microcystis may move throughout the delta. So we have 4 habitat types on our map. The main channel sites are the green dots. The flooded island sites are the pink dots. The outlets, and those are the purple dots. Those are what connects the hotspot locations to the main channels. And then static peripheral is the yellow dots or the hotspots, and those are Discovery Bay and Stockton. For our field methods, we collected 5 sediment cores to determine site heterogeneity, and we used a sample core. We collected samples in November. That's when we'd expect maximum microcystis seed stock to be on the sediment floor. So that's the end of the bloom season, but before water flows are high enough to flush those cells out. And then April, and so this is kind of the end of the wet season, water temperatures are warming, and this is the available seed stock to inoculate those summertime blooms. We did this for 2 years. So for the laboratory methods, all of our sediment samples were freeze dried. We used qPCR to quantify microcystis, looking both at total microcystis and toxigenic microcystis. Then we sequenced the microcystis genotypes and used molecular DNA fingerprinting so that we could determine if there was a relationship between these different site microcystis communities. So getting into the results, first I wanted to show you the heterogeneity of one of our sites. This is Discovery Bay. On the left is November, so when you see the maximum seed stock, and then April is the amount of seed stock available to inoculate blooms. The red is the toxigenic microcystis and the gray is total. And you can see we have wide variability depending on where we dropped our core and the amount of microcystis. The toxigenic microcystis, depending on the site, represented about 5 to 50% of the total microcystis community. And then it may look like there's not very much seed stock in April, but if you notice the scale, it's 100 million copies on the y-axis. And so we actually do have quite a bit of seed stock going into April at this site. On this figure, it shows the 2 years that we sampled. So on the left is year 1 and on the right is year 2. Yellow is sample, the maximum. So I showed you there's a lot of heterogeneity at each of the sites. I'm just showing you the maximum on this chart. So yellow is November and the turquoise color is in April. And a few things I want you to take away, as you can see on the left, we have a lot more microcystis in November than on the right side. We think that is because we had that massive precipitation event in October 2021. And I think it washed away a lot of our cells before we could get out for our November sampling. There's also that large depletion of microcystis cells over the winter, which you also saw in the previous figure. And then it's pretty clear on the right-hand side. So year 2, that Discovery Bay and Stockton waterfront have significantly more microcystis cells in April to inoculate the water column than the other locations. However, we also found that Windmill Cove and Mildred Island had some substantial microcystis seed stock in April. Okay, now we're moving on to the water column. So these, the sites are on the right-hand column, and the dotted lines correspond with when we observed the maximum amount of microcystis in the water column at each site. So first thing that I want to point out to you is these pink dots are Discovery Bay here, and then this is Stockton, this green dot. So there doesn't really appear to be much earlier recruitment at the hotspot locations than the other sites. Moving on to Julyville, Discovery Bay and Stockton tended to have a much higher bloom than the other locations. The exception is Buckley Cove. Buckley Cove is one of those outlet sites, so it's connected to a potential hotspot that then drains into the San Joaquin River. So overall, the areas with the greater seed stock tended to have higher microcystis surface blooms, and the static peripheral areas did not appear to recruit earlier, but we need more intensive sampling to fully elucidate the recruitment timing. Okay, next this is the DNA sequencing data that we did for microcystis, and this is a dendrogram and heat map of the dominant microcystis. This is for total microcystis. I don't have time to show you, but we also have a heat map for toxigenic microcystis. So what we found through our DNA sequencing is that we have 36 microcystis strains or genotypes in 2021 Delta water samples. The color change on this heat map from blue to red indicates increasing average relative abundance. So the warmer the color, the greater abundance of that strain. And what we have is 6 distinct clades. So the top clade here, this is clade 1, and this it's color coded here on the right-hand side with the habitat type and the site. So this primarily consisted of sites from main channel sites, that blue color, outlets, which is this pink color, and red, which is the flooded island sites in June, July, and August. Discovery Bay, that stands alone here in clade 2. So that's a unique strain that's only in Discovery Bay. The clade 3 fingerprints, so this was, it's right here. This is very small and consisted of rare strains from Stockton in late May and Buckley Cove, which is downstream of Stockton in early June. And this supports the some of our findings that I'll show you in a minute, that there's a potential transport between these 2 locations. Moving on to clade 4, we see a shift in the community in midsummer for the outlets, Windmill Cove and Buckley Cove. So this is probably environmental conditions driving a change in the strains that are present. Clade 5 right here is large and variable. The strains are primarily present at those static peripheral dead end sites. So this green color corresponds with Discovery Bay and Stockton. Now, knowing the location of these 2 sites, it's unlikely that there's a hydrologic connection between them. And instead, it's probably environmental conditions that are driving the strains to appear at the same time. And then finally, clade 6 here is a mix of disparate sites and strains. And the only unifier here is that most samples were collected in late summer. So next, we took all of that DNA strain data and we conducted a network co-occurrence analysis. So we're looking at the strains that were highly similar and significant to each other. And once again, they're color coded. And I'm going to focus on the water, which are the triangle symbols. The circles are sediment. And I want to point out a few interesting relationships. First, there's high similarity between samples collected at Buckley and Windmill Cove, Old River, and then the flooded island sites. The second interesting relationship is Stockton and the San Joaquin River deep water ship channel. So that's just downstream of Stockton. And then finally, there's a relationship between Discovery Bay and its outlet Indian Slough. I don't have time to show you, but we have the same analysis done for the toxigenic strains. We also have NMDS plots with permanovas that I don't have time to show you, but collectively, all of that leads to our summary that there may be 3 primary areas of cyanobacteria transport in our study region. So we documented connectivity, west side connection, Discovery Bay, Indian Slough, Old River to the flooded islands. We have a north side connection, Buckley Cove to Windmill Cove, Old River at Rancho del Rio, Frank's Tract and Mildred Island. And then an extension of that one without Old River is the Stockton waterfront to deep water ship channel, Windmill Cove, Buckley, and then ending up the flooded islands. So in conclusion, revisiting my hypotheses or our hypotheses, static peripheral areas will have the highest seed stock, and that was true. They did have the highest seed stock and are at least partially responsible for inoculating the water column during the summer. Second one are the sites, these sites are the primary sources of microcystis in the southern and central delta. There's definitely similarities of microcystis strains across these sites, suggesting high connectivity. Hotspots are likely the primary source, but they're not the sole source. There's seed stock in other locations. And these sites will experience, the third one was, will they experience earlier recruitment? They had larger blooms, but there was little evidence for earlier recruitment. So next steps, this was the first study to investigate overwintering microcystis cells in the Delta, and we have a lot more questions. We're in the process of conducting a more intensive seed stock study. Thank you to the State Water Board for funding that. And then DWR is funding UC Merced to do a laboratory-based study to better understand seed stock recruitment and test mitigation measures. I just would like to thank everyone, and my time is up. Okay, our next speaker is Keith Bouma-Gregson with the USGS. You want the pointer? Yeah. It's for 15.
All right. Hi, everybody. My name is Keith Bouma-Gregson. I'm at the USGS. I'm part of the biogeochemistry group at the California Water Science Center. And today I'll be continuing the theme of talking about cyanobacteria and cyanotoxins and looking across dry and wet water years. The biogeochemistry group and the USGS, our goal is to provide information and data to support water management in California. And so we're funded by a number of different entities. Most of the funding from the data I'll be showing today came from the State Water Board, Division of Water Rights, the Delta Regional Monitoring Program, the Delta Science Program, state water contractors, and then we have other projects funded by other entities. And so thanks to all the folks who fund our science. Some of this work was also a collaboration with EMP and Ted Flynn and that group. So I want to acknowledge all the USGS and DWR staff that have been out collecting samples that help support the science that we're doing. And also Peggy Lehman, who's now retired, but she helped kick off some of these projects in this collaboration. So the problem we're talking about are cyanobacterial harmful algal blooms in the Delta, where CHABs and Ellen and Andrea gave some great overviews of that. So I'll sort of skim over these background slides, but cyanobacteria are photosynthetic bacteria. They're present globally. They're a very diverse phylum. So there's a lot of different species. Some of them can produce toxins and bloom and have these harmful impacts. And so the three taxa that I'll be mostly focusing on today are Microcystis, Aphanizomenon, and Dolichospermum. And one of the main issues is they produce these cyanotoxins. Cyanotoxins are poisonous to humans and animals. They're classified as secondary metabolites. And if a secondary metabolite is defined as a molecule that's not required for normal cell growth, development, or reproduction. So what this means is that not all species or strains produce these cyanotoxins. So you can only know if a bloom or a sample is toxic if you test it chemically. There's a number of different types of cyanotoxins. The ones we'll be talking about today are microcystins, nodularin, anatoxins, cylindrospermopsin, saxitoxin, and annabinopeptins. These are all quite diverse molecular classes. You can see here in this figure, the molecular backbones of these molecules are quite different, meaning that they have different toxicological properties, but they also have different chemical properties in the water. Some are easier to measure than others. And then beyond just the diversity of these backbones, there's also different molecular variants within each class. Each of those can also have different toxicities as well. So the main point of that is that there's a lot of diversity, and we can't be thinking of these as just a monolithic single thing. So a little background on cyanobacteria in the Delta. The first phytoplankton surveys in the Delta were back in 1913, just over 100 years ago. And the plankton net toes there did identify the cyanobacterium microcystis. So we know it's been in the system for over 100 years. However, the first report of a bloom in the Delta occurred back in 1999, and that was of microcystis. And since then, most research has focused on the genus Microcystis and the toxin microcystin. And there's been, as Ellen just alluded to, there's these hotspots that have now come to recognize that places like Stockton Waterfront, Discovery Bay, those other coves that Ellen mentioned, have quite high toxin concentrations, whereas some of the other areas in the Delta have lower concentrations. So in spite of the presence of microcystis and these blooms for 25 years, there's no consistent monitoring of cyanobacteria or cyanotoxins in the Delta. There's been a number of special projects and research studies that have occurred, but monitoring specifically for cyanobacteria or toxins is not built into any of the long-term sustained monitoring programs in the Delta. The community has recognized this for a while, and the Delta Science Program has spearheaded an effort to put forward a strategy for what it would look like to incorporate cyanobacteria and toxins into monitoring. The draft of that strategy was released earlier this year, and one of the goals of it, the ultimate goal of that, is to implement and develop what that monitoring program could look like. So we have one of the basic questions we still have is just how do we monitor for cyanobacteria and cyanotoxins in the Delta? While we have a pretty solid understanding based off of a lot of the research and things we can glean from other systems, there are still a lot of decisions that have to be made for our system, such as where do we want to monitor, how do we want to monitor. Ellen just gave this, sort of novel idea of the overwintering stocks, and is that the type of thing we want to be monitoring for? So a lot of questions still need to be answered. Our group at the USGS, we do a lot of water quality monitoring. But beyond that, we also try to push forward the ways we monitor and try to improve our methods and our techniques for measuring the environment so that ultimately we can get good data to help us understand how our environment works. And so we were able to, starting about 2020, start to acquire additional funding for collecting samples for cyanotoxin analyses. You can see this has been pieced together by a number of different funding entities over the last few years. We have crews going out collecting water samples, so it wasn't too much effort for them to fill up another bottle. So how it's basically worked has been, we've been sampling about 18 times per year. November to April, we sample monthly, and then in May to October, we sample every 2 weeks. And this is some of the first year-round monitoring that's been occurring. Previous research projects tended to start in June or July and then end in October or November. We've tended to sample about 5 or 6 different sites. Which you can see from these maps. These have changed a little bit over time as the funding has shifted. But we just wrapped up a 2-year project funded by the Delta Science Program that was in partnership with EMP, where we were able to collaborate with them and sample these 2 EMP sites with Ted Flynn and that team. But that project's now ended. And so for this upcoming year, we're sampling these sites, which are funded, the core water quality is funded by Bureau of Reclamation, and the cyanotoxins are funded by the State Water Board. And then at each site, we've been collecting whole water samples and spat samplers. So spat samplers might be something you're not all familiar with. It's an acronym that stands for Solid Phase Adsorption Toxin Tracking Sampler. And what it is it's a adsorption resin that's sandwiched in between these meshes. We have a poster on this by Kat Cohn. Some of you may have seen that earlier in the poster session. And what it does is it concentrates dissolved cyanotoxins onto the resin during the deployment. So you can leave these out for days or weeks, and then dissolved toxins will stick to that resin. So what this provides is a time-weighted average of toxins in the system. And this is useful in flowing systems such as rivers and estuaries, where you may have a pulse of toxins moving downstream that you might otherwise miss with just an instantaneous grab sample. But the spat resin, by being in the water continuously, is more likely to pick up that pulse of toxins. Spat samplers also wind up being very sensitive because they're concentrating the toxins. So you often get detections in your spat samplers when you get non-detects in water grab samples. The downside of this is that they're considered semi-quantitative, and that's because the adsorption and desorption kinetics are complex and quite unknown when they're out in the water for those couple weeks or days. And so that does make it difficult to compare spat results directly with a grab sample. So now that we're going on to our 4th year of cyanotoxin monitoring, we thought this might be a good time to start look back, especially given the different water years that have occurred over the last 3 years. We had 2021 and 2022 were critically dry, and then 2023 was our wet water year. And a somewhat similar comparison was done by Peggy Lehman that came out in a paper published in 2022, where she looked at toxins and microcystis abundance between 2014, which was a dry year, and 2017, which was a wet year. What she found was less microcystis and fewer toxins in the wet year. And so since we also had this similar combination of a wet year following dry years, we thought this would be another worthwhile comparison to see what how the water year type changed what we found. So starting with whole water cyanotoxins, we had 6 different types of classes we were measuring. And this here is showing the detections within each class. And what you can see is we detected 3 different types: microcystins, anabenopeptins, and anatoxins. And what I'm showing you here is actually just the 2021 and 2022 data, because we did not get any detections of these toxins in 2023 due to this very wet year. And Andrea also kind of showed that in her slides. In terms of the concentrations, these were all pretty low. The microcystin and the anatoxin samples were below the California recreational warning concentrations of 6 and 20 micrograms per liter, respectively. And so These sample sites are out in those open waters. So at the same time, we had low concentrations out here. As Ellen was showing, we have very high concentrations in places like Stockton and Discovery Bay during the same sampling time. The spat cyanotoxins give us a little more to work with because we have a lot more detections because they're so sensitive. So what I'm showing you here is on the x-axis are the different months of the year. The different toxin classes are the different panels, and then the y-axis are the percentage of detections in a given month. And what I've done is I put these lowest smoothing curves over the points so you can kind of see the general trend. And what we find is that for microcystins, we start to see detections beginning in May or June. And then there was many more detections during the dry years than during 2023. And then what was notable is in 2023, we got a lot of detections at the end of the year, which I think was due to, you remember, we had a pretty dry end of the year. And also these are calendar years, not water years. So the rains didn't come till rather late. So I'm wondering if maybe that enabled the cyanobacteria to kind of persist around a little bit longer than during those droughtier years when we actually had pretty big storms roll through in October, November. In antitoxins, we actually, one of the notable things is we see more detections earlier in the year in the springtime before we really see the microcystis. And Andre also showed a similar finding. So we're thinking that there might be a different type of organism producing the antitoxins that might do better earlier in the spring. And so that's sort of like starting to produce before you then get the microcystis bloom later in the year. Cylindrospermopsins are also noteworthy because these are a toxin that we have class we haven't really detected before. The work that Peggy Lehman didn't detect cylindrospermopsins, but we are now starting to detect them on our spat samplers. So we don't really know too much about these or what organism is producing them. And so this is kind of a new field of research we need to start to embark upon. And then in general, these are just showing the detections, but similar to the whole water samples as well, we had fewer, the concentrations on the spat samplers were lower in 2023, even though some of the patterns and detections are similar. So we also have a lot of phytoplankton enumeration data collected in the Delta. So this is like microscopy. And what I'm showing you here now are these are actually the EMP stations. And so this is showing for each of the different 3 types of genera that we're looking at. Along the x-axis are the months of the year, the y-axis are the abundance of those cells, and then the bars are where those abundances within each year. And so I've kind of tallied up where these different taxa occurred. And what we see is that for Phanosomenon and Microcystis, there was a lot fewer detections in 2023, but there was an opposite pattern for Delicospermum, where we actually saw more Delicospermum last year. In the wet year. So there might be something to that wetter year. Delicospermum was also a nitrogen fixer, and also nitrogen concentrations are starting to decrease after the regional SAN upgrade. So there could also be, this might also be related to changing nitrogen conditions in the Delta. The other thing that we see is that when the detections start was in, this is really prevalent for Microcystis, but we don't really see detections in May at all, even in the dry years. The Microcystis starts to occur in June in the dry years, but it's really not till August or September here, like in these blue bars, that you really start to see the Microcystis popping up. So in the wet year, the timing of the occurrence of Microcystis is pushed back later in the year. And we also see this similar pattern. This is pulling some data from some USGS stations just from 2021 and 22, which show a similar pattern where the detections of these taxa don't really begin in June. And so these are pretty dry years in 2021 and 2022. So we might maybe start to get a better sense of what drought years look like and when monitoring could begin. Another data set that I explored was you saw from Emily, she talked about our Delta-wide mapping capabilities and the research we do. And so this is a much larger spatial extent than the EMP surveys show. And so we do mapping in May and July. And so I looked at the phytoplankton enumeration data we collect on those surveys. And similarly, in all the years, we don't really see too much cyanobacteria in May, whether it was a dry year or a wet year. But then in July, we see more in 2021. There wasn't as much in 2022, which is still a little bit of an anomaly. But then again, in 2023, we saw this similar pattern where there was much more Delicospermum in 2023 than the other taxa. And so that might maybe be indicative of some changes occurring in the system. And then the last data set that we looked at were the Microcystis visual index data. So this is just a visual index developed by Peggy Lehman, where you look over the side of the water and you estimate and you rank how much microcystis there is. Ted Flynn put together this figure a number of years ago. And this is actually data that was published through an analysis that was led by Rosie Hartman in the special issue of SFUS that came out a couple months ago. So if you haven't read and looked at all the great publications there, that is a great spot to be at. Oh, I didn't think I'm out of time, but we found that in wet years, you had less microcystis as well. And this went all the way back to 2007. So it was a bigger data set. So in conclusion, fewer detections and lower cyanobacteria in wet years. And basically by using these spat samplers and sampling year round and surveying for a diversity of toxins, we're getting a much better understanding of effective methods for monitoring cyanobacteria and cyanotoxins in the Delta. So thank you all.
Time. Another speaker used up all their time, so. Okay, now. Our next talk is by Skylar Nadelli with the USGS. Do you want the pointer? Okay.
Yeah, hi, I am Skylar Nardelli. I am a postdoc with the USGS California Water Science Center in the Biogeochemistry group. And today I'm going to be talking about the dynamics of emerging spring phytoplankton blooms at the confluence of the Sacramento and San Joaquin rivers. So as we know, the low salinity zone in the San Francisco estuary is an important rearing habitat for fish species, including the endangered Delta smelt. And X2 is often used as a habitat indicator for the low salinity zone. It's the location where the bottom salinity is 2 PSU, and it's measured as in kilometers as the distance from the Golden Gate Bridge up through the Sacramento and San Joaquin rivers. And abundances of several fish species were found to be higher when the low salinity zone was within or westward of Suisun Bay, which is marked with the black box on the map. And Suisun Bay is important because it consists of a broad complex of shoals and channels with a mean depth of around 5 meters. So a shallower environment allows for longer residence times, more light availability, allowing for high productivity of aquatic vegetation and phytoplankton. So as in a lot of systems, we are seeing long-term ecosystem changes in Suisun Bay. So this is chlorophyll A data, proxy for phytoplankton biomass near the confluence. And what you can see is there's high seasonal chlorophyll A values until around 1987 when there was the introduction of an invasive clam. And then these are lowered dramatically. There's also declines of some major fish species. And the large decrease in phytoplankton biomass in Suisun Bay is considered to be among many factors contributing to that fish population decline as it decreases food availability for the ecosystem. On a more optimistic note, over the last 2 decades, we'd seen increasing frequency of spring phytoplankton blooms greater than 5 micrograms per liter. And over the last decade, we've seen greater frequency of blooms over 10 micrograms per liter. So the potentially beneficial phytoplankton blooms could have really important implications for the food web. So the goal of our study was to investigate what the taxonomic composition of these blooms was, see if we can identify any potential drivers of these blooms, and then look at how these blooms might impact the food web. To do this, we analyzed data from 2016 to 2023. So for chlorophyll A data, we used daily average values from 2 continuous monitoring stations, CFL and HON. And then for phytoplankton microscopy, we used discrete samples collected at CFL and D10. And I will note that for the microscopy samples, there were slight differences in analysis methodology for the EMP and USGS methods. So I've just denoted which samples come from where throughout the talk.
So I'll start by sharing the community composition of the spring bloom. So these plots are showing dry and critically dry years, but I'll move into the wetter years. And each of these plots has, the black line shows the seasonal chlorophyll A from the continuous monitoring stations. And then the bars are showing chlorophyll attributed to the different phytoplankton groups. I've kind of broken up the diatoms. I've pulled out Alococera and Entomonius. I'm going to mess that up. For years, I've been calling it Entomonius, but I just learned today that it's Entomonius. So I'll probably screw that up a few times, but I've pulled those two genera out so that we can see some interesting patterns in those genera. And then any bar that has this little black arrow underneath is an EMP sample, and the ones without are USGS samples. So the first thing you might notice is that spring blooms were dominated by the diatom genus Entomonius in these drier years. And you also might notice that these blooms are occurring kind of on the early side in, mid-February to early April. Moving into the below normal water years in 2016 and 2018, you'll see that there's a shift in that peak to more mid-May. You'll also see that there are still high abundances of Entomonius in 2018 and at the beginning of the 2016 bloom. In 2016, it's a little complicated because there was a really large Alococera bloom upstream. So as that comes downstream, that dominates that chlorophyll signal. And the other important thing to note is that the magnitude of these blooms in the below normal year is a bit higher than the drier years. So we're seeing 9 and 23 micrograms per liter compared to 5 and 6 micrograms per liter. Moving into the wet years in 2017, 2019, and 2023, there really were no spring blooms, and Entomonius didn't appear to play a major role. And there is one pretty big bloom in the summer of 2017. And from upstream data, we think that that's likely transported from upstream. So what do we know about Entomonius? It has been seen prior to 2016. Here's a couple papers that highlight these blooms. It's epibenthic, so it grows on the surface of the sediments and then can get mixed up into the water column where it can form pelagic blooms. It's highly motile and chain forming, which are two adaptations that allow it for it to stay in the upper water column to get more access to light to form bigger blooms. It is found in fresh or slightly brackish environments, so that's well suited for the low salinity zone. It grows well at temperatures 4 to 25 degrees Celsius, which is a large range. So it can, opportunistically bloom in the early spring through the summer. And it also has a high tolerance to ammonium. And it's been hypothesized that this might allow Entomonius to outcompete other diatoms to form blooms. However, we looked at nitrate, ammonium, and phosphate data in relation to these blooms and didn't find any relationships. So our working hypothesis is that Entomonius filled up large seed populations on the sediment surface, on the shoals in early spring, and then a physical mixing mechanism suspends the cells into the water column and transports them from the shoals to the channels, where if there's sufficient stratification conditions, that allows for Entomonius cells to form blooms. So one of the potential stratifying mechanisms that we investigated was the development of the seasonal salinity gradient in the region as tracked by X2. Because the tidal excursion from daily average to X2 is around 10 kilometers, we focused on the region just downstream of the confluence from 60 to 75. And these plots show, so the below normal and wet years where we saw blooms. The dotted lines are the dates where X2 equals 60, 65, 70, and 75. And what you can see is that there's this uncanny timing where around 65, when X2 equals 65, you start to see a bloom occur. It hits kind of the center of the peak around 70, and then it collapses by the time X2 is 75. So it appears that increases in the bottom salinity can set up stratification that might promote these bloom conditions.
And then also the tidal mixing that is incorporated with this movement of X2 into the area can help suspend the antimonious cells to form the blooms that we saw in 2016 and 2018. So in the drier years, X2 was already above 70 kilometers in January. So that isn't a viable mechanism to start up these drier year blooms. So one of the things we looked into was tidal dynamics. And so I calculated two metrics to look at this. One is a metric that differentiates between spring and neap tides. And to do that, I took the difference between the high and the low of each tidal cycle. So bigger values represent spring tides and smaller values represent neap tides. And I also created a metric that looks at semi-diurnal versus mixed semi-diurnal tides. And to do that, I looked at the difference between the low-high and the high-low for each tidal cycle. So larger values, you have more of a semi-diurnal tide. Lower values, you have more of a mixed semi-diurnal tide. And in terms of what's good for phytoplankton, neap tides have slower current speeds, higher stratification, and a smaller tidal range, which increases residence times. And mixed semi-diurnal tides are kind of like the weak ebb flood metric that Paul Stumpner talked about this morning. So they have these periods of high stratification and increased exchange between the shoals and the channels. So linking these metrics up with the chlorophyll dynamics that we saw in 2020 through 2022. So we saw, if we focus in on when we saw a bloom, there you can see that these bloom conditions are associated with a relaxing of the spring tides and stronger mixed semi-diurnal tides, so these low values here. Both metrics are indicative of periods of increased stratification and appear to be related to these blooming conditions. So what does this mean for the food web? The first thing to think about is food quality. As far as we know, Antimonius is a high quality food source for zooplankton, so it should be good for the food web. But even if there's good high food quality, the food quantity needs to be there too. And we did see that there were lower magnitudes of the spring blooms in these dry and critically dry years compared to the below normal years. So it's possible that drought conditions are providing less food for the ecosystem. And then the timing is really important too. I mean, we saw a temporal variability from February to May for these spring phytoplankton blooms, and that could cause mismatches for certain zooplankton and fish species. So if you're a zooplankton species that's really dependent on phytoplankton blooms in the late spring or early summer, but drought conditions are shifting that bloom earlier, that species is going to be in trouble. So in summary, we saw that spring phytoplankton blooms were dominated by the diatom Antimonius, and the timing of the spring bloom was dependent on drought conditions. So drier years, we saw blooms occur earlier. Blooms were spurred by kind of a mixing and stratification event. For those drier years, it seemed to be maybe tidal, and for the wetter years, it seemed to be when the seasonal salinity gradient came into the Sassoon Bay region. And this is important to continue to study because changes in the timing and the community composition of spring phytoplankton blooms in the confluence likely impact the productivity of fisheries in the San Francisco estuary. And just as a teaser, we're starting to do some benthic phytoplankton sampling. It became very apparent to us as we've been looking at this that we don't have a great understanding of the benthic pelagic coupling, especially for diatoms. And so we're hoping to sample the 2024 spring bloom. We think it's probably going to happen in May or June based on kind of the timing that we've learned, that we've discovered. And so our goal is to characterize the broader benthic community along the shoals in Sassoon Bay, as well as the community in the water column, and then also collect a suite of environmental parameters so that we can more directly assess the drivers of this bloom. So stay tuned for that. And just to wrap up, I want to acknowledge funding from the USGS Priority Ecosystems Program and the Bureau of Reclamation. And I want to thank all the field lab and data teams that helped provide the data sets that we used here. And thank you all for hanging with me toward the end.
You teased me so. I mean, this supports all my biases, so it's great. Love it. But you pointed out that these blooms began about 10 years ago. We started seeing blooms again, but there were about 15 years when we didn't have blooms. What changed? I mean, if it's all hydrodynamics as you're describing here, the hydrodynamics didn't change.
Oh, you mean the, let's see if we can.
Yeah, like your second slide or so.
Yeah, so the big thing was that invasive clam species came in. It's
Yeah,
gonna
yeah,
take me
yeah.
a while to
That-
get back there.
That's
Is that
all
what you
fine.
mean? Or you mean like, why did they start emerging again?
Changed, which allowed the hydrodynamics to again drive the blooms, despite because the clam is still abundant, still there doing its still thing.
Yeah, I don't know. I'm not sure that we have a
I was
good
hoping you
answer
could
for that.
explain this to me. But yeah, go back to that graph because it's really.
Is there a faster way to do this?
Yeah, no,
I pass it. We're almost there.
Right there. So yeah, there's that time between 1990 and 2010 or so when we just didn't get blooms.
Yeah.
And we still had spring neap tides and we still had stratification and everything else there. What changed? I want to know.
That's D10, that's only D10. Remember, there are other places.
Oh, yeah, that is at the confluence.
Yeah, but we weren't getting blooms anyway.
There were blooms in 2000. In 2000, there was a bloom 30 micrograms per liter in Sassoon.
Yeah, that's it.
Yeah.
Yeah.
Yeah. Still a bit of a mystery, but we're looking into it.
Solve it for next year.
Thank you. Okay, we have one last talk. Yeah. You want the pointer, right? Do you want it? Yeah. Oh, is it yours?
No, I think that's still good.
No, sorry. Like a new computer next time. Okay. Our last talk is Nick Framstead with the USGS. We have a slow computer.
Last talk of the day, so thanks everybody for sticking around. I'm Nick Framstead with the USGS California Water Science Center, and the last couple talks touched on sediments, and Ellen's talk was all about sediments, so they teed this up really nicely. I'm gonna be talking about sediments as a nutrient or source or sink to the Sacramento-San Joaquin Delta. So the basics of why care about nutrients. Nutrients form the base, or they fuel the base of food webs through giving the next essential fuel for primary producers such as phytoplankton or macrophytes, floating vegetation. And then these go on to support higher trophic levels. And sediments are a large pool of nutrients because this is where all of the organic matter accumulates in aquatic systems. So potential mechanisms that can release sediments from the, or release nutrients from sediments are redox potential near the sediment water interface, bioturbation from organisms within the sediments, or just sediment resuspension from mixing. So there are a few systems where sediments are critically important to the nutrient cycling. A local example is Clear Lake, and in Clear Lake, 70 to 95% of the total phosphorus loads to the lake come from the sediments underlying the water column. So that's basically all of the phosphorus is coming from the sediments. In Lake Erie, 60 to 200% of the external ammonium loads come from the sediments. So sediments can represent very important sources of both nitrogen and phosphorus to aquatic ecosystems. I'm not saying the delta is like these systems, but there are conditions that set up in the delta that can mobilize sediments or nutrients from the sediments to the water column. Traditional methods of measuring sediment nutrient fluxes are these core incubations, and they're great, but they do have their limitations. One of them being container effects. where the initial conditions in one of these sediment cores might be a little bit different than another, and these can propagate over time and cause drastically different conditions between these cores. That's not due to the treatment. And it's also difficult to recreate in situ conditions in the lab. So there's some issues with some of the traditional methods. So something my group did, an experimental approach, was bring the lab to the field, where we deployed a chamber that created an enclosed system over the sediments. And we used this system to track nutrient fluxes over time within this chamber. So we basically had a chamber that was placed over the sediments, and we recirculated water within that chamber using pumps through our flow-through system, which has a bunch of in situ instrumentation that measures a lot of water quality parameters. And that way we can get change in nutrients over time within this system. We also injected a bromide tracer to get an estimate of the volume of the chamber, so that we can then relate the concentrations to actual mass fluxes from the sediments. It's a pretty crazy system, and unfortunately, I don't have enough time to get too in depth into it, but we're gonna dive into this study results. So the design was we collected a bunch of sediment cores throughout the delta at the locations shown, 24 paired sites, and these pairs are wetland and deep water or channel sites that are adjacent to each other, so we can get an estimate of habitat type. So it's about 60 cores. And then we subset these same sites again in the summer of 2020. So we got cores from 2020 and 2021. And we measured a suite of carbon, nitrogen, and clam content that we'll get into later. The next thing we did was take the actual benthic flux measurements using that chamber system that I mentioned earlier. And this was all done during the summer of 2021. And we did 18 sites, 13 that had repeated samplings within that same summer. And then we did these at paired wetland and channel locations. And I'm not sure if the colors are coming through, but the pairs are all colored together within this map. There we go. So first of all, diving into the sediment characteristic results, the sediments were highly variable within the delta, anything from inorganic sand to very organic rich peat. And this was here is percent nitrogen and percent carbon shown in these two maps. And we wanted to look at broad general landscape trends in these variables. So here we can see that in the southeastern portion of the delta, more in the San Joaquin portion, we see higher percent nitrogen and higher percent carbon relative to the rest of the data or delta. And this makes sense because there are high nitrogen concentrations in the water column in this region due to all the agricultural inflows, and that can also make itself show up in the sediments due to settling. And then the high percent carbon might be due to vegetative history of this region, wetland vegetation being in the sediments, or it could be detrital phytoplankton that have sank to the bottom in this region due to high phytoplankton biomass. Then moving on, so a couple of our outlier sites where one of them was peat and it had the highest percent nitrogen. This was near Frank's Tract. And then this other site had very low percent nitrogen. That's because that was just pure sand. So that's just an example. Moving on to carbon and nitrogen ratio and Olsen phosphorus concentration. So we had the highest carbon and nitrogen ratio again in this peat site. We don't see the same broad general trends spatially, but we do see that adjacent sites can have drastically different carbon and nitrogen ratios. So perhaps this is more habitat dependent. And then Olsen phosphorus, it's actually covered up, and I need to figure out how to jitter the points a little better, but the highest site was near Fern Island with Olsen phosphorus concentration of 160 micrograms per gram sediment. And then we also, another important variable in this study that we didn't expect was clams. The clam density varied quite a bit. We see that there's not especially broad trends, spatial trends, but the highest densities were at these two sites, one in Ulatis Channel within the Cache Slough complex, and then the next one was down here. And they had densities of around 2,500 clams. This is just number of clams, not biomass, per meter squared. Looking at these same variables by site type or habitat type, we have channel open water sites in the purple with wetland shallow water sites in green. We see that generally wetlands had higher percent nitrogen and carbon than did the channel open water sites. Same with phosphorus and chlorophyll. And again, this chlorophyll is just on the surface of the sediments. So this can be either epibenthic periphyton or just settled phytoplankton. Another striking difference is that clam density was way lower in our wetland shallow water sites. And this is not a comprehensive study of clam densities, but it was a noticeable pattern. These three outliers are all from one site near Ryer Island in Sassoon Bay. The rest of the sites in our wetland sites had next to no clam density, which is important, and I'll get into that later. So looking at our benthic flux rates, we have dissolved oxygen, DIN, which is the sum of ammonium and nitrate. We have flux rates for all these. You can see all of the DO fluxes are negative, meaning that DO was consumed within the chamber. So that's what we'd expect because this chamber was dark. So microbial respiration and respiration from plant matter, whatever, was driving the consumption of DO, which is good. Should be that way. Our chamber worked in that respect. We also noticed that sediments are sources of DIN, mainly in the form of ammonium at 9 of the 12 sites that we have data for. Sediments did not happen to be a major source of nitrate, so I'll plot nitrate and ammonium on the same axis just to show the difference in magnitude of these two fluxes. And then another noticeable pattern was that the same site could switch between being a sink where sediments were taking up nitrate to a source where nitrate was being given to the water column at the same site. And these were sampled in June and August. So if there's repeated samplings at the same site, we did see it flip-flop sometimes in terms of nitrate. But in general, ammonium fluxes were more consistent at a single site. Plotting these out in a map, we don't have the same spatial resolution to make any spatial inferences, but we do see that immediately adjacent sites like here in Sherman Lake can have drastically different DO fluxes. So it's likely that this is due to differences in habitat type or sediment quality. DIN, we see that the highest DIN flux was here at Ryer Island in Sassoon Bay. Again, no crazy spatial patterns popping out. Ammonium patterns followed the same as DIN since most of the DIN coming out of the sediments consisted of ammonium. Nitrate, again, no crazy broad spatial patterning, but again, adjacent sites can have drastically different nitrate fluxes. So looking at these benthic flux rates plotted out by site pairs, in which these sites are two different habitat types, but they are immediately adjacent to each other within the delta. So we're trying to control for landscape influences, but just analyze habitat influences on these flux rates. We see that there is a noticeable trend between pairs in terms of DO fluxes. So wetland sites can have higher magnitude or like more dissolved oxygen consumed relative to the channel open water sites, which makes sense because wetlands probably have more organic rich sediments, a little more microbial activity taking place than in the deeper channels. So that's interesting. But we don't see the same consistent trends between DIN, ammonium, and nitrate between our site pairs. So we're not seeing that habitat type is as strong of a predictor of these nitrogen species fluxes from the sediments. And these are just examples of a more wetland-ish site. And again, this is a very subjective classification and a channel open water site. And getting back to clams, since I mentioned them earlier, so clams did have a significant correlation with DIN, ammonium, and nitrate flux rates from the sediments. And there's a mechanism for this. Potential mechanism is through nitrogenous waste excretions from clams. So if they're giving off nitrogen or bioturbating where they're stirring up sediments at all into the water column, that could be a possible mechanism. However, they had low R-squareds for these correlations. So there's likely a lot of other factors that are at play in determining nitrogen fluxes from the sediments rather than just the presence of the density of clams. But they're important to pay attention to when trying to calculate benthic flux rates of nitrogen. So in summary, delta sediments are highly variable, anything from sand to peat. Sediments are likely a source of DIN, mainly in the form of ammonium. And this can be an increasingly important ammonium source in light of the wastewater treatment plant upgrade on the Sacramento River, where ammonium inputs were cut by almost 100%. And DIN inputs from that point source were cut by about 2/3. So this ammonium source could be increasingly important within the Delta. And neither habitat type nor landscape influences fully explain flux rates from the sediments. It's probably a complex interaction of sediment characteristics, residence time, flow, stratification being able to set up at these sites. And this approach was pretty time intensive, but we would like to include replicates at a single site at the same day in order to gain a better estimate of the uncertainty associated with this method. Regardless, this data is valuable for modelers, and we have capabilities to collect more. And with that, I'd like to thank my colleagues at the California Water Science Center for collecting all this great data, and the Delta Stewardship Council for funding this study, and IEP for holding this workshop, and take questions if we have time.
Thanks, really interesting. I'm curious what you would expect to find in a bed of SAB. So many of these habitats are not unvegetated, and so I'm curious about that.
Yeah, and I probably should have talked about that more. There was some SAV present at some of the sites in our study, and that is a very good question. Something we'd like to explore more, maybe in Frank's Tract in areas with a lot of SAV. How do they affect fluxes? But it's also hard not to disturb it when we put this chamber down. So you can slice open, plants, and that can release nutrients as well. So, but yeah, we'd like to figure that out.
Could there be interactions between the clams and other variables, like, if you're putting the chamber down on clams on like a nitrogen-rich substrate, that you would, have an interaction effect that could be quantified?
Yeah, like in terms of clams preferring sediments with higher nitrogen content,
or mobilizing the nitrogen, only if it's, rich in the sediment that they happen to be in.
Yeah. Yeah, that's a good point. Yeah, we could test sites that have low sediment nitrogen content, but still have clams. Yeah. Yeah, that would be a very interesting analysis. And it seems like it's an important question of what is the role that clams play? Is it active or just associated with other things? Great question.
Do more. Sorry. So we're at the end of the session, but feel free to come ask the speakers questions if you want. Thank you all for attending.
And that concludes day one. Thank you all the presenters for excellent presentations today. Thank you all for staying to the end of the day, and we'll see you tomorrow. If you're not going to be attending in person tomorrow, just please drop your name badges in the bucket up by the registration table so we can reuse them. But I'm sure you're all coming back tomorrow, because tomorrow is going to be as good as today, if not better. So yeah, have a lovely evening, and we'll see you tomorrow.
If anyone wants